So I will start directly with the introductions for today.
This is correct.
So Nora, what we're going to do is we're going to have Carol give us a recap, and then after Carol, if you want to make some initial comments, I think that would be great. Is that okay with you, Nora?
Absolutely, yes, of course.
Okay, everybody, thank you for joining. It's good to be with you for day 2. I'm going to now turn everything over. Just remember, for questions, please use that Q&A box. And then you can, if it's for a specific person, please add that person's name. All right, we're going to turn everything over to now Dr. Eklund.
Thank you. Welcome, everyone, to day 2, Enhancing Health Disparity Research Related to Substance Use and Addiction, Research Gaps and Opportunity. We'll begin the program at this time, and Dr. Carlos Blanco will begin our agenda today. Welcome, everyone.
Yeah, so thanks, and welcome, everybody. Yesterday was a very, very successful day, a lot of ideas, very robust discussion, very satisfying. And today we have also a very, very exciting program. So whereas yesterday we focused more on the social aspects, today we're going to focus more on the basic science aspect. Probably many of you joined us yesterday, but for those of you who didn't have a chance to join us, we have asked Dr. Carol Hubner from the Division of Therapeutics and Medical Consequences to give us a brief recap of yesterday, and that should set us up for today. So Carol, whenever you're ready.
Thank you very much, Carlos. First, let me say that yesterday's keynote speakers and panel discussants provided a wealth of information and insight into ways NIDA can enhance its portfolio on health disparities research. So in my very brief summary, I'm going to be highlighting some of the concrete steps that I heard discussed, recognizing that these are just first steps and that much of the more work will need to be done. So if I could have my first slide. Great, thank you very much. So as I said, these are going to be organized according to recommendations that were made by our panel discussants and speakers. The first of which is the need to increase funding of community-based research so that NIDA's grant portfolio better reflects its commitment to addressing racial inequities in substance abuse treatment. What was mentioned, and which we all can agree upon, is that the environment impacts behavior, and the environment clearly influences vulnerability to substance use and treatment outcome success. Dr. Alegria spoke eloquently about the idea that treatment and research must be connected to the communities where people of color live. And what does that actually mean? For me, it meant, and I think for many of us, it meant that community-based research allows for the direct investigation of those factors that not only create, but perpetuate health disparity. There was a robust discussion of many of these factors. I'm only going to mention a few. They have to do with questions about and issues about employment and working conditions, housing, the availability of healthcare, and particularly within this country, the availability of medical and health insurance. There were also important discussions about barriers to post-treatment community reintegration, as well as the policies and laws that perpetuate health disparity. It was also discussed that NIDA needs to be committed to developing strategies to mandate equity to improve health disparities and intervene on social determinants of health. And for me, the big take-home message about this recommendation is that NIDA's ability to balance its portfolio towards greater inclusion of community-level research is going to go a long way in improving substance use treatment outcome in underserved population. So that leads me to the second recommendation that was made, and that has to do more with study design. The idea being that it will be important that we recognize the validity of different clinical study designs, particularly when we're talking about studies addressing racial inequity. So we're all aware of randomized clinical trials. And the charge was, in essence, we need to start thinking beyond that. Clearly, RCTs are the gold standard, and they have an important place in clinical research. But there are other study designs that are careful not to control for the contextual variables that are the actual drivers of inequity. As a result, when we use these kinds of designs, we're actually studying the factors that are important. Another a recommendation tied to this was that we need to include racial minorities beyond ticking the box. And I think, unfortunately, part of my slide is cut off. But the idea here is, if we're going to call this racial equity research, underrepresented populations have to be the focus of that research. It's not just saying they're part of the study design. The purpose of the study design is to investigate and evaluate their challenges. If I could have, well, excuse me 1 second. So how do we implement these kinds of recommendations? And that brings me to the next slide. The third recommendation that was made by the group yesterday was that NIDA should work where possible to implement changes to the scientific review process. The purpose here is that we should try to encourage addiction research that flows through the social science lens. And what does that mean? Well, one way that can be accomplished is to go beyond what was called champions on study section. And I just think that was a very kind way of saying that we need much more racial diversity in study sections. We can't have just 1 or 2 people who are there to be the voice of underrepresented populations. But that's not sufficient. It was also recommended that NIDA look to people with research expertise that is outside its traditional portfolio of reviewers. What does that mean? Well, the recommendation was social scientists. Some people talked about economists, urban planners. These are people who are doing racial health disparity research, but are looking at it from a different perspective, a perspective that it was suggested that NIDA might benefit from. They're the kinds of people that can look at certain applications and say, from my perspective, this is important. And that's something that was highly recommended to improve the funding of racial equity grants. Diversion, equity, inclusion training for reviewers, that was also something that was discussed. It's important because what it does is it signals to reviewers NIDA's priority in funding racial health disparity grants. It's putting it in the forefront of their minds. When you're reviewing grants, keep this. This is a priority by NIDA. We want you to attend to this. And I should say, it's something that I should have mentioned from the beginning. This was all outlined in Dr. Verholden's talk. I pulled almost everything out of that presentation. She also talked about something that I wasn't familiar with at all. And that is when reviewers are evaluating the productivity of PIs, it might be best if they would look at the approaches differently for PIs that are submitting community-based research. What does that mean? Well, the normal metric is number of publications. That's measure of productivity. That might not be appropriate for PIs submitting community-based research. Maybe what they should be, their research should be based on or measured by is what impact does it have on the community? What is the work that they're doing going to do to improve the lives of people dealing with substance abuse issues in the community? And so I thought that was fascinating because that's a different way of looking at PI productivity. And finally, the last recommendation had to do with funding set-asides for disparity research. And here again, I want to point to a term coined by Dr. Verholden, which is if we want equity, we need to mandate equity. What does that mean? Number one, including benchmarks for funding applications for minority serving institutions. Here we're talking about historically Black colleges and universities, tribal colleges and universities, and the like. And NIDA has a good record of setting aside training grants, but there was an emphasis in the discussions that it should go beyond that. What we want to do is support programs of research that are submitted by underrepresented minority scientists. And I'm, excited to repeat what was presented yesterday by both Will and Carlos, and that is Dr. Volkov's commitment to set aside funds for disparity research. There has been a commitment of over $100 million over the next 10 years that will be dedicated to funding research to reduce disparities and inequities. And with that, I want to thank you for listening, and I will hand this back over to Will. Carlos.
It's okay. Will and I are of one mind, so one can cover for the other. And we work together very well. So thanks, Carol. That was a very, very good summary. And now what we wanted to do is ask Dr. Volkov, Nora, if you have any additional comments as we start our journey today.
Well, I actually thought that the summary was excellent, and unfortunately, because of network connection problems that we had at NIH, for those that have Verizon service, I was thrown out, so I couldn't listen to all of the presentations, but these gave me a very good perspective of some of the most salient issues. And I do think, I was resonating very much with the concept of mandating equity, and I think that in terms of how do we translate that into actionables? And there are some suggestions that were given there that I think would help us. And definitively having benchmarks is, in my view, to the only way that we can accelerate equity on research and on knowledge that will get rid of the health disparities that we are all very aware we are currently and have been living. So I thank you very much, Carol, for that excellent summary. And now I want to hear the presentations. I'm very interested. It's a fantastic program. So Carlos and Will, thanks again for your organization.
Thanks, Nora. So I will turn it to Will, and I think Will is going to introduce the first moderators on the first panel. So Will, whenever you're ready.
Perfect, great. Thank you, Carol. Thank you, Dr. Volkov. So we're going to switch gears today. Yesterday, we talked a lot about social determinants of health. Today, we will focus primarily on harnessing basic science to understand racial disparities and the impact of racism. We hope at the end of today, we will have some discussions on the integration of social determinants of health and basic science. So that is something that we hope to challenge our group today to have some discussions and recommendations on. So without further ado, I'm honored to introduce our first presenter, our first keynote presentation by Dr. Zaraza, who is the Associate Professor, Department of Psychiatry and Behavioral Sciences at Duke University Medical Center, who will present on genomic architecture, brain science, and neuroethics. Dr. Zaraza, welcome and thank you.
Well, thanks so much for having me here. Let me go ahead and screen share. I'm assuming a quick thumbs up if everyone can see that okay. All right, great. I am Kafui Zaraza. I'm an assistant professor or associate professor of psychiatry and behavioral sciences, biomedical engineering, and neurobiology at Duke University. And if anyone has any thoughts or questions that they can't get in during the panel, feel free to write me at Kafui Zaraza on Twitter. So I am a psychiatrist, as I mentioned, and I got really interested in basic research as an enterprise to bring healing to my own family. So my family has been deeply impacted by illnesses like schizophrenia, depression, and bipolar disorder, many of which are, of course, comorbid with substance use disorders. And in thinking about the pathway to ultimately come up with treatments and cures, one of the most exciting discoveries for myself and my family over the last few years has been the discovery of what we begin to say is the genomic architecture of schizophrenia. And I'll sort of point out at this point in time that I'm a basic science researcher. So I work primarily with mice, but my hope that the talk today will show you how the work that we do in basic research laboratories ultimately connect with the human and clinical and translational enterprise as they move forward. So this is just a slide. It was a study that was done, a genomic study in which they were looking at the whole genomic architecture of individuals with schizophrenia. And the question here is, are there genes that that give rise or risk to schizophrenia when you compare those to the genes of folks who do not. And in this study, they were able to find 108 associative genes. In other words, 108 marks or hits in the genome, which give higher risk for schizophrenia. So this was, of course, extremely important for me in the work that I think about my lab. What I would basically do is take one of these genes and then study how it changes the brain function of a preclinical species or model organism like a mouse. Now, one of the things that's really important to appreciate about these studies that have looked at genomic architecture, this is a study of over 100,000 individuals. And I never used the word exclude, but what I became aware of was, if you look at this study of over 100,000 individuals, there was no one of African ancestry in these studies. And I don't like using the word exclude, but it's, from a simple math perspective, it's hard to appreciate how a study that large would statistically not include anyone of African ancestry. But this is certainly not the case simply with neuroscience research or mental health research. On the right, I'm just showing you a slide looking at our understanding of genomics and the risk of gene changes and how they're linked to disease across time. And what you can see is that these studies are dramatically overpopulated with individuals of European ancestry. And when I use the term ancestry, it's basically how have your genes coursed across space in the world over the last 500 years. So these studies, even though 16% of the world is of European ancestry, the vast majority of information that we know about how genes are linked to disease risk are of individuals of European ancestry. And this has really important implications for how we think about human disease. Here, I'm just showing you, if we look across the architecture of how genes, your gene profile, so the complicated or complex profile of genomic differences that you have based on your ancestry, how much does that architecture give you an ability to predict disease states? And I'm just showing you how that data that was originally learned in individuals of European ancestry extrapolate across populations. In other words, how much does this apply to individuals of different ancestry? And you could see in the group of individuals with African ancestry, the architecture that was learned in the individuals of European ancestry fails to extrapolate. It extrapolates really, really poorly in individuals of African ancestry. So I'll shift for a bit and go back to the work I do as a basic scientist. So I'm interested in how brains encode emotions, and I record the activity of a lot of cells in the brain, and I figure out how they pool information together. And I use a set of techniques called machine learning, right? So I use complicated, really high-powered computers to find patterns in a lot of data. And so I'll give an example of what that looks like. Imagine we had a task here. I've got a bunch of O's on the screen, and the task is to figure out what the line of symmetry is in the O's. So I might use my machine learning algorithm to determine the line of symmetry, and I learn a really nice line of symmetry for a bunch of these sample O's, these 6 sample O's. And then in order to show that I have done this correctly, I would go to a new population, in other words, a new group of O's, and I would see how much my line of symmetry extrapolated to this new group of O's. And you could see that I've done quite well. Then I might say, well, let's make sure, we want to be sure before we start developing clinical therapeutics that our line of symmetry is a good one. So let's go to a new population and see how much it extrapolates again. And again, we've found that we've done a really good job, right? So this is the idea of training a model or determining what the line of symmetry is and making sure that it extrapolates. All right, now we want to get a little more complicated. So we've got a mixed population of I's and O's. We want to know how much that line of symmetry extrapolates. And again, we do a really, really good job. So far, so good. So we develop our clinical therapeutic, and then we apply it to a broader part of the population. We've got some W's now. We apply our line of symmetry, and all of a sudden it fails. And what you appreciate here is that the line of symmetry that we learned on the I's and the O's does not extrapolate to the W's. And what you'd really want to do in an ideal situation is that you'd like to include the W's, the I's, and the O's all in your training set. And the reason why this is particularly important is because when you do this, you actually learn a different line of symmetry, right? So there still is a solution. It's actually a different solution that you would have found if you only included the O's or you included the I's. And now when you go to a broader population, you find that this line of symmetry extrapolates again. So this is the real world example that links to the genomic picture that I showed you previously. So I'm just showing you a slide here. It's from my colleague at Caltech, Viviana Gradenauer. And I'm going to tell you about a tool she's developing with some real thoughts about how to use this to treat illnesses in humans. And she's essentially developing a tool that will allow us, by doing injections, systemic injections, to be able to target new genes and proteins to cells in the brain. In other words, we can turn on and off and change activity in the brain in a way that's therapeutic, in a targeted way. So when we think about our medications that we give, they can activate and target lots of brain cells. She wants to target brain cells in a very precise way using these compounds. And what these compounds will actually do is the viral compounds will cross the blood-brain barrier and target different cell types. And so she's developing this tool, and she started off using a bunch of different strains of mice. Now, we can all argue that these are, and appreciate that these are all mice, right? So within the same species, they're just different strains, so slightly different genetic differences in the mice. And what she found when she was developing this tool was that this tool crossed the blood-brain barrier really well in some strains of mice, the ones researchers we tend to love, the C57 black 6 mice, you see that on the left, but it didn't work well in other strains of mice at all. The fluorescent color shows how much this tool has crossed the blood-brain barrier. So in one strain of mice, it works really well. In one strain of mice, it doesn't. So you can imagine the challenge that we would run into if she developed this for this first strain of mice and then tried to apply it more broadly to the population. What she then did was she went through a series of additional viral tools. This is different versions of the tool, PHP-B, PHP-C2, PHP-C3, so a bunch of different versions of the tools. And instead of just testing them on one strain, she tested them on the 2 strains that were most different. And what she ultimately discovered was a specific tool in this preclinical setting that worked across the most strains. And so that is the one that she's taking forward, because she's ensuring that she has what is essentially the best line of symmetry as she's developing the tool, not waiting till afterwards to test the tool more broadly in different strains of mice. So why is this so important, and why would I choose to lead off my talk around neuroethics and genomic architecture in this way? Well, as many of you all know, we are in the middle of the Federal Brain Initiative, and the goal of this initiative is ultimately to develop new tools and technologies that will allow us to both understand how the human brain works, but also to create a platform for treating devastating illnesses, things like depression and Alzheimer's and drug addiction, by creating these new tools and new therapeutics. And so this is currently underway, funded to the tune of about $4 billion, launched in 2013. And so I want you to imagine a world where we have tools, these viral tools that can cross the blood-brain barrier and deliver treatments to certain types of brain cells. And then we think of all of this new tools and new type of science being done. This is just an example showing you where they're showing that they can actually target viruses to specific brain cells. And this works in preclinical models, things like mice, and in humans and non-human primates. And so we can now develop tools which will cross the blood-brain barrier and target different cells. And we can show that we can actually make a potential tool like this work in humans. So imagine now we're trying to put these two things together. One, a tool that will target specific brain cells, and then an atlas, which will give us the code for each of the cells that we're trying to develop. And this is what we're doing in the Brain Initiative, getting this atlas that tells us which cell, what is the code for each individual cell that we should be turning on and off. And I'll sort of frame this because I think this is particularly important. As I mentioned in the genomic architecture case, these large-scale patterns, so these tools that we're developing, the identity of genes matter a lot when you think about ancestry. So like I said, with the polygenomic architecture, it worked in individuals of European ancestry. It didn't work so well in individuals of African ancestry. So a bunch of us have been thinking about, as these new treatments are being developed, we want to make sure that they work for individuals of African ancestry and other ancestries as well. And we don't simply want to wait till the end. We want to make sure that this works in the beginning. So this is a team of individuals. We're actually organized by Dr. Reverend Al Hathaway. He's the pastor of the Union Baptist Church in Baltimore. And he made the point that he wanted to ensure, as these technologies were being developed, that the African-American community, particularly that in Baltimore and more broadly, wasn't left on the outskirts. He wanted to make sure that individuals in the community were participating in the clinical trials, and that we were also thinking about the genomic differences that might exist as we were coming up with these new treatments and technologies. He was able to pull together with Eddie and Sylvia Brown, who are entrepreneurs in Baltimore, and they funded the beginning of what we would call the African Ancestry Neuroscience Research Initiative, also partnered with the provost of Morgan State University to ensure there's an educational component there, and the Lieber Institute, which is a nonprofit brain research institute in Baltimore as well. And as this initiative was being launched, a bunch of people approached Dr. Hathaway and pointed out, how African Americans had been treated in the biomedical research enterprise. And they also wanted to make the point that they didn't want to participate in a study that would actually show that Black brains were worse. And so the community has been involved in this since the beginning. The communication piece has been there. And I'll just show you one bit of preliminary data that's come out of this, and then I think you'll understand how important an effort like this is. This is just looking at human brain tissue. So the Lieber Institute had about 700 brains of African ancestry, and they just did initial analysis of these human brains now, looking at gene expression in different brain regions. Around the rim, you see the different chromosomes. And then you see 3 brain areas. In the center, you can see hippocampus, and then in the middle, you see an area, dorsal lateral prefrontal cortex, particularly implicated in illness, and then around the rim striatum. And wherever you see a red dot, that is a gene that has a higher expression profile in the brains of African ancestry. And wherever you see a blue dot, that is a gene that is higher expressed in individuals of European ancestry. And so the point here is that if you look across the brain, there are different gene expression profiles that are related to the ancestries. So I'll pair this now with the work that I showed you around Viviana Gradinaru's work. You can appreciate now that if you're using gene expression as almost a GPS system to target cells in a disease case, that if you do not generate algorithms that figure out how to find the points of symmetry across ancestries, you could have a tool which activates an area that would be helpful, like dorsolateral prefrontal cortex in one ancestry, that then activates another area like hippocampus that might be really harmful across another ancestry, such as individuals of African ancestry. So I'll conclude this talk just by putting up a picture. I've been reflecting on this with it being Black History Month. And I'll ask you just to sit for a moment and think about what a slide like this makes you feel. So what are you experiencing? Perhaps it gives you some sense of discomfort or anger. You think a lot about what people have experienced through the history of this country and the adversity. And even the central theme today is about how we bring more equity to the biomedical research enterprise. But when I see this picture, what always stands out to me is that I'm an engineer. And so I see what happens when technology is selectively applied to one group of people while the other group of individuals is left behind. And when I look at this picture, what I ultimately see is this. And so my hope is that as this initiative, the BRAIN Initiative, is being developed to create a platform in which we'll be coming up with new tools and devices to treat human illness, particularly around addiction, the basic studies and the platforms we're using to include humans and understand the architecture of gene expression and brain cell types will be sure to include individuals of African ancestry as well. So thank you so much for having me here.
Well, thanks very much, Avi. That was an amazing talk, going from symmetry of the letters to the brain to bathroom. I mean, in 10 minutes or 15 minutes, I mean, very didactic. I think it's really fantastic. So we don't have a lot of time for questions, and so I'm going to move directly to introduce our second speaker, which is Dr. Jean Brody. So Jean, whenever you're ready, thanks for agreeing to be part of this fantastic meeting. Take it away.
Drs. Ackland and Blanco, thank you so much for involving me in this important meeting. I've learned so much from all of the participants, and it's given me a lot to think about. Today, in my own presentation, I'm hoping to raise awareness about the value of embedding biomarkers in longitudinal epidemiologic studies and prevention trials that will enhance our understanding of the biological impacts of racism and other social injustices. All of the data I'm going to report today were obtained from different samples from rural African-American families. They are representative of families who live in what's known as the Black Belt, that area of our country that starts with North Carolina, South Carolina, travels from Georgia, Mississippi, Alabama, Louisiana, and a little bit into Oklahoma. One of the characteristics of this part of our country is that 35% of the nation's poor live here, and over 60% of the children raised here live at or in poverty. What's also another characteristic of people who are raising children in this region is that they are often largely invisible in their towns, in their cities, in their states, and for the most part, they've been invisible in science. Dave, next slide, please. In this first study, we're going to look at a biomarker called allostatic load. And what we did is from 500 young adults, we collected overnight urine voids from which we assayed the stress hormones, cortisol, epinephrine, norepinephrine. We drew blood to get biomarkers of inflammation. We obtained blood pressure, body mass index, and we aggregated those in a well-known way, in a cumulative fashion, and that defines allostatic load, which forecasts all kinds of cardiometabolic diseases, strokes, and cancer at midlife, and it also forecasts early mortality. So in this first, what I consider just a proof of principle study, we tested the hypothesis that adolescents exposed to frequent encounters with racial discrimination would forecast higher levels of allostatic load. And we also tested a second hypothesis known as the parent effects hypothesis that's observed in animal models wherein positive caregiving tendencies exert lasting effects on offspring physiology, in the brain, in the neuroendocrine system, and the immune system. So what we did is across ages 16 to 18, we developed and obtained lots of data on encounters with discrimination. Through some statistical procedures, we were able to create two groups of young people, those who encountered high and stable levels of racial discrimination, and others who encountered low and increasing levels across their adolescent years. And we also obtained across ages 16 and 18 the kinds of emotional support that these young people received from their parents based on their parents' reports. So the figure shows that at age 20, young people who had frequent and stable encounters with racial discrimination across ages 16 to 18 showed higher levels of allostatic load at age 20. And this association was not significant for young people who received high levels of emotional support from their families during adolescence. So there's support here for both the idea that exposure to high and chronic levels of racial discrimination does have some effects on dysregulating physiologic systems that forecast later disease, but not for everyone. And there's some support here, or strong support for the parent effects hypothesis seen in animal models. Dave, next slide, please. In this next proof of principle study, what I wanted to demonstrate was how across 2 samples, exposure to high levels of racial discrimination would be associated with forecast accelerated cellular aging. And we obtained a marker of accelerated cellular aging from epigenetic analyses derived from whole genome assays. So in this first study, we had a sample of young people who once again provided data on their encounters with racial discrimination across ages 16 to 18 years. And we also obtained from their parents the amount of support that they received. And the data was really clear and actually stronger than we expected. The highest levels of accelerated cellular aging were found among young people who were exposed to high and stable levels of racial discrimination, but not for all of them, once again. Some of these young people who were exposed to high and stable levels of racial discrimination were shielded from the cellular effects of racial discrimination because they had access to supportive caregiving relationships. Dave, next slide, please. So we wanted to replicate this in another study, and what you might notice at first blush is the data looks so similar. So this is a second sample. The youth in this sample were ages 17 to 19 years old. Once again, we obtained data, this time at age 22, of accelerated cellular aging. And we also obtained data once again from the parents of these young people across adolescence on their receipt of supportive caregiving. And the results, as you can see, mirrored the first set of results, with the highest levels of accelerated cellular aging at age 22 were from young people who received high and stable levels of discrimination and lower levels of emotional support from their caregivers. The reason that we focused on accelerated cellular aging is that, like allostatic load, it forecasts the chronic diseases of aging later in life, and it also forecasts early mortality. Next slide, Dave. One of the things we do in addition to studying the etiology of drug use and health disparities is we use some of the information we collect in longitudinal epidemiologic studies as the fuel or the active ingredients in prevention programs that we develop for rural African American families. And these programs are developed in close collaboration with the rural African American community. And we've been fortunate to develop 3 efficacious programs that were designed to prevent the onset and escalation of drug use. The first program is the Strong African American Families Program for pre-adolescents. The second program is the Strong African American Teen Program for 15-year-old young people. And the third program is the Adults in the Making program, which is designed to offer some protection for young people as they go out into a world that has limited educational and occupational opportunities. We've embedded some biomarkers in some of these programs, and I'm going to share with you as proof of principle analyses of things that other people might think about in learning and using clinical trials to inform the effects of racial discrimination and other risk factors on the development of young people. Next slide, Dave. So this is a new study. And I thought it was very germane. In two of the prevention programs, the Strong African American Teen Program and AIM, we had baseline data on exposure to racial discrimination. And what we wanted to find out was, could participation in a prevention program that enhanced supportive caregiving ameliorate the effects of racial discrimination on increases in symptoms of mental health problems? So if you look at panels A and panels B, you'll see that young people in both of those trials who were exposed to high levels of discrimination 2 years later showed increases in conduct problems if they were assigned randomly to the control conditions, but not to the they didn't participate in the prevention trials. If you look at panel C, which focuses on depressive and anxiety symptoms, you'll find that for those participants in AIM who were 17 years old at baseline, those young people who experienced high levels of racial discrimination at baseline and participated in the control condition evinced higher levels of depressive and anxiety symptoms across 2 years, whereas there was no differences between the other three groups. Dave, can I see the next slide, please? In the rural Southeast, as I mentioned earlier, poverty is very prevalent. And in this analysis, what we wanted to find out was, would participation in a prevention program, the SAFE or Strong African American Families Program, offer some protection against growing up in poverty on neurodevelopment. And what we did is we simply counted the number of years young people lived in poverty and looked at the volumes of the left amygdala and 2 areas of the hippocampus to ascertain whether we could find some protective effects of supportive caregiving. And the data depicted in slide A, in panel A, clearly show that participating in SAFE did protect against declines associated in amygdala volumes from living in poverty over the course of adolescence. And similar findings were replicated for hippocampal areas. So together, these findings show that effects of prevention programs that promote and enhance supportive caregiving have effects that are detectable in the brain at age 25 after experiencing the prevention program at age 11. And what I'd also like to say is we have found similar effects on other biomarkers 8 and 14 years later. And those biomarkers include stress hormones, norepinephrine and epinephrine, pro-inflammatory cytokines, cellular aging, and metabolic syndrome. So we think there is a lot of merit in embedding biomarkers to start to get at what is it about racial discrimination and correlated variables that have implications later in life and can be protected with participation in family-centered prevention. And I want to finish by saying all of our family-centered prevention programs were designed to deter drug use and risky behavior. And because they're one of the central mechanisms in those programs was the enhancement of supportive caregiving, we found effects on health outcomes that were unanticipated at the time we designed the programs. Thank you.
Okay, thanks very much, Dr. Brody. That was also a fantastic talk. We are going to do now is take a very short break until 2 o'clock. And so we will reconvene at 2 o'clock to start the panel discussion. So we will have a few minutes, but we will start sharp at 2 o'clock. All right, thanks.
Integration of race in the basic science research to better understand health disparities. This session will be moderated by Holly Moore, Program Officer from the Division of Neuroscience and Behavior. Thank you, Holly.
Thank you, Will. And first, thanks again. I'll echo the thanks to the meeting organizers, Will and Carlos, for such a really different meeting. It's been wonderful to listen to everything so far. As we jump into discussion, I will remind attendees that you can use the Q&A function to submit questions or comments to participate in the discussion. I think on behalf of the panelists for this session, or the headlining panelists, I want to invite panelists from the other sessions to feel free to contribute, jump in. You might use your Zoom hand raisey thing to jump in, but if I miss it, just go ahead and feel free to jump in. And I'll start out, I'll introduce our panelists as they jump in. And I'll start out with just introducing the a priori questions we had for this session. The first question was, how and why should basic scientists incorporate race into their studies? Another question was, what research designs and methods are appropriate for addressing questions about race and racial inequities in health? What are the implications for recruitment and community engagement with regard to people who might participate in research? And what steps or considerations should we take to support or address limitations in data interpretation when trying to make our research relevant to health disparities, social determinants, and basically addressing health inequities in general? So Raul Gonzalez from Florida International University offered to break the ice and jump right in. Raul is a professor of psychiatry. I have you here, sorry. Psychiatry, psychology, and immunology at Florida International University. He heads the substance use and HIV neuropsychology lab. He comes from a clinical neuropsychology background, and his research is aimed at how neurocognitive differences across the lifespan may impact risk for substance use disorder and other behavioral disorders. So Raul, why don't you jump in with your response to the first question?
Sure. Thank you, Holly. Definitely want to thank you for organizing all this. And I have a lot of gratitude to express to all of the speakers from yesterday. I've learned so much and feel incredibly privileged and grateful to be a part of this session. So the question is, why should basic scientists incorporate race into their studies? And this is a question that I've been grappling with. It's a question that folks on my team have been grappling with. One of my hats is a site principal investigator for the Adolescent Brain Cognitive Development Study, which is for those of you who don't know, a very large study following around 12,000 youth starting at age 9 to 10 until age 20. It is a study with a lot of variables that are being looked at. It has a very strong neuroimaging component. It has genetics. it has neurocognitive functioning, and so many other social variables tied to that. One of the things that I was asked was to give a little brief introduction to myself, which I think is going to segue directly into part of answering this question. In addition to being a clinical neuropsychologist who doesn't typically work in this space, I'm also an immigrant. I was born in Cuba. Technically, I think that makes me a refugee instead of an immigrant. When my family came here, I was 5 years old. As I was growing up, my parents would tell me that my race was Cuban. And by the time that I got old enough to check off check boxes on forms, by the time I was 5, I learned that I was actually Hispanic. And then when I moved to San Diego for graduate school, I found out that I was actually Latino. And then about a decade ago, I learned that I was actually Latinx. And very recently, I've learned that I'm actually BIPOC. And my 23andMe ancestry suggests that my ancestry is, all sorts of other things. My wife was born in the Dominican Republic. Her stepfather was also born in the islands. He was born in the Dominican Republic, but grew up in Trinidad. In Trinidad, his race was Dominican. And when he moved to the United States, he became Black. And several of his children born in the United States have grown up and identify as Black. When my wife moved to the US at age 16, she hated checking off those check boxes. I was already pretty much acculturated into the check boxes by that point in time. When she was given an option, she would always look for other, and she would check off other and write in Dominican. By that point, I was then shocked. I was like, Sarah, do you know that you're causing a data analyst somewhere a tremendous amount of grief by checking off these open, check boxes and writing stuff in? But that did not dissuade her. Our daughter was born in Chicago, and we moved to Miami when she was starting preschool. And in preschool, they had, during the first year, an activity about where are you from? What's your heritage culture? She came home and asked us, and we explained to her what it meant. And she says, okay, she processed all of this, and she said, I'm Chicagoan. So her whole presentation at school was around Chicago-related stuff, because that's how she identified. And her own identity is also developing along the way. So race is a socially constructed, imprecise, flawed, and fluid construct. It's inextricably tied to a social, cultural, and geographical context. So how does it help us to better understand the human condition? Should basic scientists incorporate race into their studies? A 1992 JAMA article, so we're going back a couple decades, by Osborne and Fate, invoked the Hippocratic Oath and said, do no harm in publishing studies of racial differences. Write about race in a way that does not perpetuate racism. In 1999, the Human Genome Project concluded that there's no evidence for race as a biological variable in the genetic code. Another JAMA article in 2003 by Kaplan and Bennett provided guidelines for the responsible use of race and ethnicity in biomedical publications. And they implored that there should always be a thoughtful reason for the use of race in any study, that they should not be used as proxies for genetic variation, and that in their interpretation of any sort of race results, all conceptually relevant factors should be considered. Yesterday, we heard from Dr. Hansen, very powerfully and very clearly identifying race as a hierarchical taxonomy that is part of our master narrative, a master narrative in this country that is clearly harmful to many people. We heard Dr. Verholden articulate that there's no reason for race to explain differences in addiction, but plenty of reasons that implicate discrimination, racism, and social determinants. In my own process of learning more about this area and space, I've focused so much, like so many of us do, on the very narrow work that is at the core of what we do, and sometimes ignore folks in other areas like anthropology who've been grappling with these questions for a very long time. So this very long answer, I'm going to wrap up with quoting from an article that was published in 2005 in American Psychologist by Smedley and Smedley, and I think the title of it really sums it up completely. The title is Race as Biology is Fiction. Racism as a Social Problem is Real. And I encourage everybody to read that. So this is a direct quote from this article. From its inception, race was a folk idea, a culturally invented conception about human differences. It became an important mechanism for limiting and restricting access to privilege, power, and wealth. The ideology arose as a rationalization and justification for human slavery at a time when Western European societies were embracing philosophies promoting individual and human rights, liberty, democracy, justice, brotherhood, and equality. The idea of race distorts, exaggerates, and maximizes human difference. It's the most extreme form of difference that humans can assert about another human being or group, as one of its components is the belief that differences are permanent and cannot be overcome. This article goes on to say that racialized science, with its emphasis on identifying immutable differences between racial groups, can be expected only to maintain and reinforce existing racial inequality, and that its adherents indirectly argue that no degree of government intervention or social change will alter the skills and abilities of different racial groups. Yet, policymakers cannot ignore the fact that social race remains a significant predictor of which groups have access to societal goods and resources, and which groups face barriers, both historically and in the contemporary context, to full inclusion. The fact of inequality renders race an important social policy concern. So, should basic sciences incorporate race into their studies? I don't have any great insights or answers. It's complicated, but the more I think on it, the more it becomes obvious to me that it's time to move on. And we have to do better.
Thank you, Raul. Next, I'll introduce Natalie Slopin. She's a social epidemiologist from the University of Maryland, whose work focuses on social influences on health and health disparities, and psychological and biological mechanisms through which childhood experiences can impact health, risk for disease. And Natalie, would you want to jump in just to respond to Raul, or actually address any of the other questions?
Sure, yes. I will plan to build on Raul's first thoughts, sticking with this question of why should basic scientists incorporate race into their studies. And the answer that I had come up with, with these prompt questions in advance dovetails really nicely from where you left off in your conversation. So from my perspective as a social epidemiologist, I would argue that it would be very helpful for us to be reframing the question to ask, why should basic scientists incorporate experiences or features of the environment associated with racism into their studies? And so, as you cited study after study, there's not evidence for biological or genetic differences between these groupings that we so often use in the United States for race and ethnicity. So we need to ensure that our studies are designed to focus on the systems and structures and experiences that differ between these groups based on the social and environmental characteristics, factors related to wealth, educational quality, access to healthcare, air quality, etc., so that we have this broad representation of the factors that we truly believe are driving the health disparities that we observe. And so while these categories of race and ethnicity are fraught with problems and imperfections, they still are important as a tool. They're the best we have at the moment to monitor and use these socially constructed categories to ensure that we're moving towards equity and closing gaps in health that do exist. And so we are at a point in time where I would argue we want to continue to include assessments of aspects of the environment that are associated with race and ethnicity, and ensure representation of those checked boxes that you referred to in your talk, to make it so that our studies are incorporating the populations that we need, and that this is going to be really important for developing policies and interventions to effectively address health equity and improve population health.
And lastly, I'll just reintroduce Kaf Jazi from Duke. As you saw, the cool research that he does and thinks about, he uses different genetic tools to manipulate psychiatric risk genes in mainly mouse models or rodent models, and then uses really cool ways of manipulating circuits to figure out what the functions of those genes might be and how that might play out in a psychiatric disorder. And Kaf has also been at the forefront of discussions about race and ethnicity with regard to inclusion, as you saw today, also how to actually improve race diversity in the scientific workforce, and also things like that actually are not unrelated, like data dissemination, tool dissemination, things where we want to think about how there's a gulf actually between basic science and how it actually does or does not help the kind of science that actually has real impact on delivery of healthcare to people. And he's part of that conversation as well. So Kaf, can we get your thoughts on what you've heard so far?
Yeah, I mean, I'll sort of follow up with both of my colleagues, and I will agree that certainly race is not a biological construct. But I'm both a neuroscientist and psychiatrist, and so I certainly appreciate that external environmental variables are certainly manifested in how our brain matures, it shapes itself, and which genes it chooses to turn on and off in a way that is undoubtedly behaviorally relevant. So while I will 100% agree that race is not a biological construct, it certainly is, and I'll say presumed race, is a social construct and through which society mediates environmental effects that certainly have a lasting impact on brain function. And as I can certainly say, as a Black male, I can certainly say that like racism is stressful, y'all. Like this last year has been incredibly on top of everything else, from watching the disproportionate deaths of COVID-19 to much of what we've experienced more broadly in society and academia, like the effects that racism has and how that is mediated undoubtedly is it's stressful, and undoubtedly it has an effect on the biology of the organ in my head, no doubt about it. So I do think there are ways that we need to be thinking about these clear environmental effects, both of one could appreciate as a basic scientist, but one can appreciate this as a psychiatrist as well, and figure out how to shape some of the communication around these studies being done. I mentioned some of the work we're doing in Baltimore. And one of the things you can appreciate in Baltimore is you sort of like move from block to block, like the life expectancy is like 20 years different from block to block to block. And there's no doubt that that is certainly reflected in the, what we'll say, presumed racial demographics that exist from block to block to block. And so there's no doubt that there's a link there. Now, I mean, I would argue fairly, I think, to say that the causal mediator is redlining. In other words, there's a history of separating people by race. And there are things that are more likely to show up in some of the homes than others, like lead paint, which certainly has developmental impacts on the brain, which certainly shapes health outcomes. And that is undoubtedly associated with presumed race, but that is the selection that is used to mediate those racist effects. So I think in order to begin untangling that, we have to begin to dig into directly what the structure of power is and what racism was used to do. It's why when we're building this African American Neuroscience Research Initiative, the first thing was to build the community, right? And it was the first pillar has to be the community. It has to be Reverend Hal Hathaway that I showed his picture of, because you know he was talking to folks and he's like, look, we have brains already, but before we engage in doing neuroscience on brains of African ancestry, what are your concerns? And the community is like, look, there's a long history in which people have been arguing that Black people aren't as smart, from phrenology to everything else. And how are we going to participate in an initiative that brings about the outcomes that we're interested in, but doesn't create additional evidence for people to say, see, we told you, there's evidence now that there's differences of function of groups. So we think about the communication and the community involvement up front. There's a clear neuroscience motivation for this, as I pointed out. If I'm taking genes and putting it in mice and doing basic neuroscience research, I want to make sure we're picking the gene patterns that are going to extrapolate to the broadest number of humans, not just those of European ancestry. But in order to even begin to do the basic science work, I'm not agnostic to the fact that the work I do in a mouse is relevant to humans, and humans need to be involved in shaping that enterprise more broadly. Not simply because, I'll end my soapbox shortly, not simply because, you know, we live in a democracy and our health enterprise is funded by our population at large, therefore they have to be bought into the science and it has to benefit them, but because, you know, we as scientists certainly should appreciate that, you know, we're not the simple and only stakeholders and beneficiaries of the wonderful institution that is NIH. So there's an obligation for us to make sure that as we're discovering things, if we have two set of choices, one is a scientific paper and the other is a scientific paper and the maximum benefit for the community, that we're thinking about how to do the latter. I don't always think it's a choice community or scientific progress. We can certainly make choices that deliver on the second as well. So thank you so much. And I'm excited to participate in this panel.
Thanks to all three of you. I mean, one thing I'm hearing is that, first of all, those boxes that Raul talked about, the boxes that are checked with regard to race and ethnicity, they don't have a scientific history. They weren't generated with any scientific evidence behind them. They're not just imperfect. They're actually they're actually a proxy for a system that we really don't want to perpetuate. And it's going to take some time for us to change the conversation about what race means with regard to how to do research. And one of the things I think about when we talk about genetics, for example, is that probably the study of the effect of racism on genetics and how people have been changed by racism across generations is more scientifically valid than the topic of racial differences in genetics. And with regard to race, having a phrase that's an alternative or a different phrase, or basically not using the phrase race differences, but rather talking about the different stressors and the different challenges faced by different people on the basis of what others think of them as others think of their grouping. So it's going to take a lot of conversation to kind of tell each other what we mean by the construct of race, and that's something that's going to take some time, and we might talk about that a bit more. The other thing, I mean, I'm also going to sort of like go with some things that people are contributing on the Q&A and contributed before the meeting. And one of those is relating to research design. So given that we have this conundrum, given that when we say, well, we need different racial and ethnic groups in research, yet we know that construct is imperfect. How are we going to do that in a way that we remain sort of fluid so we can change and improve the construct such that we're just including everyone that needs to be included in health research and we're eliminating health inequities? How do we eliminate health inequities? But in the meantime, we still have to use these constructs to go out and reach populations that have been underserved. Maybe Natalie, start.
Sure. So I think that's a great question. And in my view, I think it's really important to be thinking about characteristics related to our sampling, the measures we use, and as well as the study design. So first, thinking about the sample, I'll make 4 points. So I'd say first, it's important to think or to design our studies for some of them to be focused across racial and ethnic groups, and some to take an in-depth look within particular groups, ensuring that we have heterogeneity across these important social constructs that we believe are really important to health. There's a lot to be learned from both these cross-group comparisons and in-depth studies of single groups. And it's really important to avoid systematic biases in the way that we sample, which can have major implications for the generalizability of what we find. And we also want, related to this point, is we must ensure that we have variation in the socioeconomic position within groups so that we can make meaningful comparisons. It's very common for our cohort studies perhaps to not do as good of a job as this, which would be ideal. And then we end up with a lot of confounding where we're maybe misattributing differences or we can't explain racial variations in certain outcomes when we could have, should we have had more representative or more heterogeneous samples where we could more accurately characterize the range of social environments and capture what's driving health. I also want to make the point related to samples that in the United States, there are certain groups that are basically systematically not studied, or perhaps in our national surveillance data, this data is being suppressed and not released within the public release files because these groups are small in size. And it may be a privacy issue of why they may not be disclosed, but because of this, we're lacking really important information for understanding disparities that exist. Related to this, we have large groups that we may have available to analyze, such as, well, not even large, but we know, for example, that our US data often groups together Asian Americans. And this is an extremely heterogeneous group that can be hard to interpret based on the distinct social experiences for people who are arriving in that category within a data file that we may be working with. So data disaggregation is really important for exposing gaps in health equity, and something that we should be keeping in mind as we develop our sampling approaches and continue to move forward the agenda. And then finally, related to sampling, there's been increased importance of capturing notions related to intersectionality. So, the experience of individuals with multiple marginalized identities within the way that we are modeling our health outcomes and disparities, because we know that outcomes can differ dramatically based on different combinations of stigmatized attributes by our society. Related to measurements, we know from social science literature that when stressors are measured comprehensively, we do a much better job accounting for racial and ethnic variations in health and health disparities. And so there are stressors that are common across all individuals in society. And then there are certain types of stressors that are much more likely to occur among individuals of certain racial and ethnic groups, such as Blacks and Hispanics, that may pertain to both interpersonal and institutionalized forms of racism. And so we need to be thinking about the assessment of stressors very comprehensively in our work. And then finally, and not only comprehensively, but at multiple levels. And then finally, from a design perspective, I want to make the point that a life course approach is extremely important. So we know that both risks and resources accumulate over time, and that these matter for health and substance use outcomes. So there's increasing attention recently to considering adversity in childhood in relation to substance use, particularly among Black and Latino youth. And this appreciation of life course and trauma are really important both to our etiologic studies and also for the development of effective and trauma-informed interventions. Thanks.
Thank you. Anyone else want to jump in on that? One thing I'll interject real quickly before you, so I'm going to retract that, Raul. I'm going to jump in. No, just to say that one thing that I want to highlight or reinforce that Natalie said was the importance of recognizing heterogeneity within a group that's identified on the basis of, again, this false construct of race and ethnicity. But we have to actually include the heterogeneity within those groups if we're going to actually get a full picture of how health inequities arise and how we're going to address them. So go ahead, Raul. Sorry, I interrupted.
Sure. Just want to, there's a couple things that have been said that I want to jump on. Dr. Jaraza's talk was fantastic. I think there were several very key points that were driven home that resonated with me. One is the importance of having that broad representation of people in a human subject study. And then Dr. Slopin has also talked about this importance of having that within group variation. So with ABCD, which is a study that's taking place across 21 sites, the original goal of the study was to have a representative sample and using sampling techniques so that we can match the US Census and maybe oversample some groups. But boy, it sure is hard and expensive to do that. And it sure is hard and expensive to do that with a large enough sample size. And at the end of the day, I gotta say the efforts were Herculean. We didn't quite get there. At first, we did not have enough individuals of Black folks in the study, and NIDA stepped up. More money was given to increase recruitment efforts. So this is part of the equity process of maybe it takes a little more to get to that representation. But then we couldn't find enough whites with low education that wanted to participate in research. And then we missed out on some of those folks as well. And then, as has been pointed out, so what do we use if we're not using race, which is a nice lazy measure to try to get a bunch of this stuff in, and it still falls short at the end of the day? We want to look at income. We want to look at environmental toxins and environmental exposures, quality of education. I mean, the list goes on and on. Discrimination, racism that's experienced, stress, all of this. Again, the price tag keeps growing and growing and growing in terms of the feasibility of doing some of this right. So there needs to be a lot of resources pumped in for us to do this right. And there also needs to be some sort of gatekeeper effect. One of the things that we're struggling with ABCD is it's an open science study. So we are collecting data on all of these variables, but what's to keep somebody from using these variables in a racist way, or to continue to perpetuate the master narrative using the data that we're collecting with such great intentions? So I was humbled and shocked when I was going through the literature of looking to see how other folks have dealt with these issues, to read papers that were written 3 decades ago on how to responsibly use data on race, and seeing how little that's trickled into the work that my colleagues and I can do. So what do we need? Do we need journal editors to start putting very clear rules on how this works? ABCD has a lot of great folks working in it that are putting a lot of thought into these very questions. There's talk about putting language in the data use agreements for ABCD, and I hope that this comes to fruition, about explicitly not using race as a biological variable and for sorting people out or for making kind of racist conclusions of this group's brain is bigger than this brain. And look at how it's impacting us negatively. Dr. Jaraza brought the point home. I mean, what were people scared of? People didn't want to participate in our research because they're afraid it's going to get turned around to continue perpetuating these harmful ideologies. So it seems that there needs to be racism, there needs to be some sort of gatekeeper control about how this data can be used. And within ABCD, I know a lot of folks have been talking about preparing like a rapid response team when a paper gets published with ABCD data that misuses this type of data to shoot back commentaries on how the data was misused. So yeah, I don't think we have any answers, but it requires a lot of support. It requires support from leadership. It requires support all the way up to NIDA. And this workshop that's been happening over these 2 days goes a long way. And we were talking earlier in the chat about how so many folks feel that just these couple of days are required viewing for a lot of folks in doing the type of work that we do.
Kof, did you want to contribute there?
Yes, certainly. I'll start off by saying I think it's really important, and this is a lot of the importance of the biological framing, is to point out that ancestry does matter, right? So I don't use the word race, the term race in that matter, but I do use the term ancestry, right? So ancestry is certainly how our genes have moved across environments over time. And as a basic scientist, I can appreciate this. I'm not allowed to call a mouse unless I back crossed it 10 generations, right? So certainly there's a lot of relevance in terms of how our biology interacts with the environment that is linked to our ancestry. And our move to think of everyone the same, which is important, should not be lost as we're developing a precision medicine initiative, which says that every single biological organism, which we as human beings are, every single one is actually different in a way that might be meaningful for health. So I think it's important to always keep a flag post there that we are trying to understand individual architecture in a way that's relevant for treating disease, and the individual shouldn't be lost in that. At the same token, I'll say, certainly I appreciate that a lot of movement has to happen from where we are now to where we're ultimately trying to get. And it's going to require a lot of investment as well. But I do think there are things that we could do immediately, and they're small and subtle shifts, but they're likely, you can imagine that they would have a huge impact on the field. So I was doing a thought experiment as I was sitting here, and a great friend of mine recently, who is a radiation oncologist, so she spends a lot of time thinking about physics, pointed out, she said, objects in motion tend to stay in motion unless acted upon, right? And I think a lot about this idea of race and how it's percolated our scientific enterprise. And you can imagine how small and subtle of a shift it would be if instead of saying race, we said likelihood to be the subject of systemic racism, right? Which is likely the exact same thing, right? And we just shifted it that way. And all of a sudden, what you're describing in studies is a thing that the entire system is doing wrong to somebody, right? And I can imagine how easy it would be for somebody to sign up to a study and it's like, oh, I get to prove that there is systemic racism and it is actually doing something wrong and is having a difference in health outcome. And so all of a sudden the framing, right, around how we think about, how we interpret and how we communicate race, instead of saying it is, it is likelihood in the society that we exist in to experience a set of factors that causes a different outcome. And then certainly one could do the follow-up studies and say, well, the mediators are income and how lending practices may change how one moves through society, or student loans or multi-generational stress or police brutality in a community. But we frame it from the broad perspective of that's actually what we're talking about. And that's what the larger impact is of the health outcomes that we're observing.
Thanks. And yeah, just to extend on that a little bit, heritage is it's a nice term to start with kind of like to also think about the idea that social systems, of course, impact heritage. Basically, everything a generation faces, everything a generation goes through affects how they pass their genes and other basically heritable things down to the next generation. And across generations, if that forms a heritage, that will certainly be impacted in a major way by the social systems surrounding people of that heritage. And so thinking sort of hearkening back to yesterday, trying to, even at the level of the most basic science, think about the impact of social systems, or at least thinking about the relevance thinking about a research scientist question about biology, at least in the context of social systems that may have affected heritage, might be something that is interesting to kind of think through.
Yeah, and I think that the idea there is certainly saying if we don't have a long history in which race is used poorly, then we can sort of become neutral in how we're navigating it, right? But the idea here is actually to create almost an anti-racism counteraction to say, and this is how you've thought about race. The counterbalance to that is actually to say race has actually been mediating a negative effect. And I think that's really important for framing even how we as academics tend to view that more broadly across the enterprise.
Along those lines, there's an interesting sort of study design question that's coming from one of the attendees. And I think it actually has a lot of like many layers of an answer. And that is, how can we or how do we measure racial discrimination in our research subjects? What are the different ways we measure racial discrimination or the effects thereof?
I'm happy to jump in. I'll tell you a little bit about what we're doing within ABCD, which has been an evolving, growing process. The kids are now about 13 or 14 years old. Some of these different things that we're talking about kind of come into play at different stages. I remember when we were getting started with ABCD, a lot of folks were saying ABCD is an imaging study. ABCD is all about the brain. But very early on, we had the liberty of forming these different work groups that focused on other relevant and important constructs. And a lot of folks were brought to the table. And in retrospect, there's a lot of things that happened that I think can help us. So for example, we are asking our participants how much discrimination they've experienced. Have you ever been discriminated against because of the color of your skin, because you're an immigrant, because of your body type, because of your sexual identity? We're asking if they feel discrimination from teachers, from other people in their lives. So we have this self-report aspect of it. We also ask youth about and the parents about their heritage culture and about participating in activities of their heritage culture and other cultures. We ask about ethnic identity and how they identify. So in addition to the standard checkboxes, there's, and we go back a couple of generations as well. So this is all within the ABCD data for folks to hopefully use this in a very positive and responsible way. with these sorts of variables. So this is in addition to all of the biological variables that are being collected. There's also collection of baby teeth for youth, where some of the other investigators are trying to get information on the environmental toxins. So some of the things that I've heard so far about what could be done in study sections, this bringing in reviewers from other adjacent relevant disciplines into the review process, sounds like a great idea to me, because we all have blind spots as very focused, narrow researchers. What I heard from Dr. Gerasa about the folks that he brought in from the community, not just to kind of be advocates for the study, but to listen to what it is that they have to say that needs to be done for the research to make an impact in the community, is something that I'm also taking to heart, because it's critically important. The part that concerns me is the availability of resources. I mean, ABCD is such a Herculean study with so much resources behind it. I want to be also pragmatic and think not everybody's going to have the money and the funds that go along with the research that they're trying to do to be able to collect data on all of these variables, even if they had the best intentions. So we have to come up with, if we can't do it right, then we need to be upfront when we write our papers about how flawed the methods that we're using are, to not inadvertently communicate that there is something magical, static, and immutable about any sort of racial differences that we see. I mean, that could be a start.
Natalie, I see you raised or nodding.
Yeah, so I will build after Dr. Gonzalez's response to say, in addition to these individual types of surveys of people's interpersonal experiences of discrimination or ethnic identity and that sort of thing, I know that ABCD and many other of our federally supported studies nowadays collect the participants' addresses so that they can geocode characteristics of the social environment to characterize the social structures, the quality of the environment in terms of toxins, and other ways that individuals are going to differ based on the types of opportunities that they have for healthy development, for high quality education, and all of these things that we know drive disparities across the life course.
I've wondered also, particularly yesterday when we were talking so much about social systems, if that the idea of bringing different kinds of science to the table, particularly areas of science that aren't typically funded by NIH. So really bringing very different kinds of scientific perspectives to the table when it comes to study sections, journal editorial boards, and things like that. It might help us prioritize the variables in some way. It is the case that we don't have enough resources to basically proliferate variables to collect about people in a non-prioritized way. But I'm wondering if there's a way of the different kinds of science to weigh in on how variables can be prioritized, or whether or not there's the constructs that we're using to create variables are valid across different disciplines.
Yeah, so I can I'll sort of diverge a little bit, but I think this will come back to the topic that we're on. So one of the things that I think is critically important is not just thinking about how to expand study sections and expand participants in research, but we need to be thinking more strategically about how to expand the participants of the researchers, right? So those that are actually asking the questions and participating in the enterprise. Certainly, as we look at the whole NIH portfolio, 1% of the scientists NIH funded in the extramural program are Black, 5% are Hispanic. And as we move towards 2050, where the majority of the country is going to be in a racial or ethnic, what we consider a minority group, we haven't done a good job of figuring out how to extract those ideas and talents and perspectives of that research group. So I think there needs to be strategic focus on those efforts as well, right? Some of how, we can certainly postulate on the best strategies to quantify things. And, it's essentially hypothesis generation. The way we solve this is we have a hypothesis and we do the study and figure out the best way to quantify, what these factors are and how to think about these things in population population such that we can capture them and ultimately understand health outcomes. So we really need to be thinking about how to bring scientists into the enterprise, allow them to ask the unique set of questions that come along with them being an individual and coming from a trajectory through growing up here in the United States or migrating here that otherwise has not classically been funded and supported in the research portfolio of those asking questions. These things are particularly of importance focus now. I know there's a lot of attention been paid to funding disparity differences, particularly for Black scientists. I just think it's time to not simply think about the topic choice, but the diversity of talent in our country being brought into the enterprise as well.
Yeah, I think one of the questions that does come up with regard to the scientific workforce issue is, is this question of not just bringing in, not just changing the demographics of the scientific workforce, but finding a way to change the sort of master narrative, change the kinds of questions we're asking, and allow the new faces that we bring into the pipeline to be basically the forces behind this change. So I don't want to basically help young scientists come in to the pipeline and drink the same Kool-Aid that I drank. I want them to change the narrative.
Holly, I have to tell you, the young scientists, I mean, they're holding our feet to the fire. I'm so grateful to them. It's incredibly uncomfortable at times, but it's necessary for change to happen. What I've seen from some of the youth coming up now, they are brazen and unapologetic and they see change as attainable and they want to make it happen. And I wholeheartedly encourage all of them, despite whatever reactions they tend to get from their PIs, have some patience with us, bring us along for the ride. And I'm certainly grateful for them. They see things differently.
I want to jump to a topic that I don't want to miss before we leave, and that is a kind of maybe outside of the box. I hate those phrases, but out of the box thinking, or an honest discussion about what we typically think of as basic and translational science, where it fits into a health equity mandate, or and that is, if I define basic science as any science with any kind of population, any species that's just designed to uncover a fundamental principle underlying, that's actually conserved across living organisms. That's basic science. Translational science is actually using those principles, trying to look at basic fundamental principles as they might apply to disease states or disorders. A lot of times, scientists in these areas, particularly those scientists that don't use humans, oftentimes just see themselves as separate from the conversation of health equity. And I'm wondering what your thoughts are on how we're going to better bring those scientists into the conversation in terms of what they need to know, how they can better and maybe more humbly interpret their data, what they have to contribute. What are your thoughts on that?
Yeah, I'll say for me, I was extremely challenged in that. And you could appreciate this from the research talk that I gave. I'm a basic scientist and I think about, genetic manipulations and study how they affect brain cells, right? And, it was when I appreciated, again, my family suffers from profound psychiatric illness. And when I appreciated that the genetic manipulations that I was recreating in mice that were based on European ancestries. Right? It was quite the wake-up moment for me for thinking about how me as a basic scientist was so upholding an enterprise that was less likely to yield cures and therapies for my family on the other side of it, right? So I think sometimes we think of this in such a distinct and separate way, right? Like it's a mouse, right? Like if its whiskers go a little bit slower, why is this relevant to human health? But certainly the knowledge that we create has the potential to extrapolate in ways that that we need to be aware of more broadly. At a minimum, in terms of training scientists, we need to be aware of this in terms of how we're thinking about and training that next generation of really awesome scientists, right? Like I honestly thought of myself as being a young scientist until I went for a run and I was super sore the next day. And it occurred to me that I was like over 40 and needed to start thinking about the trainees as the next group of scientists and making sure that I create space for people to bring a new set of ideas and perspective into the field as well. So even at a minimum, those that are sitting at the bench thinking primarily about a protein or cells or flies need to be really aware of the role that we are playing in creating the next generation of those that will ask critical questions. And you mentioned peer review as well. The reason one must be so, it's so critically important to be aware of systemic racism and the innate biases that humans have is that we all have them, right? And we have an innate bias towards, recreating self, which, we could live with if the history of self was even and equally distributed in this country, which it certainly isn't. So I think it's really important for everybody at every part of the enterprise to realize that we are not operating independently and that the decisions and choices we make have a profound potential to ultimately shape the health outcome of individuals.
And along with that, before we go to, I'd like everyone to jump in there, but we have a mandate for in training for responsible conduct of research. That is a component of training for any NIH funded training program or fellowship. Is there value in having training on health inequities and social determinants of health as simply a part of the basics responsible conduct of research curriculum, in addition to responding to what Kop said?
Yes.
Can I? So this is Tali Baron.
Go ahead, Tali.
Yeah, I'll be part of the next panel, but I was really intrigued by the depiction of, or the perhaps definition of translational basic science, because, we give things definition just like we do with race, and sometimes it might be kind of helpful to think about what the whole concept means. There are many different ways to look at basic science. Basic science, you can think of, if you want to know how the TV is broken, you absolutely need to know how it works. And we will never figure out how the brain, for example, functions without going in directions we never knew existed and exploring. And this can be basic science. But again, we always know that whatever we discover can be broken, and that will be a basis of disease, including a propensity or vulnerability to substance disorder. In terms of translational science, again, what we have here is oftentimes a clinical question that we cannot answer in human or in a clinic, and we take it to the lab to try to understand it with the understanding that that may be relevant to a given human disorder or not. But the translational really is bidirectional. You go from the clinic to the lab and back. And yes, the bridges are sometimes a little fraught and wet and snowy, and it's not entirely clear what aspect of the clinical questions we need to ask and what aspect we can actually address in basic science. If we call basic science the use of experimental models. I think that it might be helpful to really kind of blur the boundaries. We are interested in understanding, the normal brain and the normal reward circuitry, and then the propensity or vulnerability or action or interaction of the brain and drug use and drugs to the brain across a spectrum, a repertoire of different types of brains, different ages and what have you. So I would really vote for not necessarily picking out basic science or translation science or clinical science, but really considering them as a continuum. And if there is an important question, such as the biological impact of racism or other types of adversity, they should be asked across all of these, the various spectrum of science, with each part of it contributing its unique assets and strengths and powers, and clearly understanding that some questions cannot be asked in a given set of experiments.
An idea of, instead of categorizing research, just ask people, what is your question and what design do you use to study your question? That might be the question we ask each other.
What organism, what model, what set of tools, what aspect of the question are you focusing on, rather than putting it in little silos of basic translational and clinical.
Others?
Hi, this is Ron Palmer. I'm in the next panel as well, Tali. I just want to second what Tali just said. I mean, I just came from a talk that I gave on cross-species data integration. And we were asking the same kind of questions. We were not discussing about the impacts of race, but everyone is considering how diversity plays a role in how substances impact organisms. And there are researchers who are trying to create diverse panels of organisms that we can begin to understand the impacts of substances and how best to translate that. So I think what we're all thinking about is the agglomerative effect of all of the science that informs our understanding and how best to develop treatments.
Thank you. John Loon?
So I want to go back to what Dr. DeGrasse was talking about in terms of how ancestry is very important. I think one of the things that I think a lot of us are doing is, in the rush of trying to avoid the discussion of racism, we're trying to say, okay, let's go beyond. And I think Raul was guilty of that. I think let's go beyond what happened. I mean, there's a history from 1619 to present day as a young black American myself, even though I'm an immigrant, I grew up here. And I'm assuming that Kofi went through the same difficulties. And it's very difficult to go beyond these, no matter how much we say, wow, the country needs to be better, these stresses stressed our brains and actually educated the kind of questions that we have to deal with all the time. So we're not ready to go beyond those issues. We need to discuss them clearly, honestly, and not obfuscate about the presence of racism. And ancestry does matter. And I think we should pay attention to those. Anyway, that's what I wanted to say.
And I think part of the part of what we were saying is that, and sorry, but that maybe the conversation just needs to sort of change the perspective in terms of the construct of race and ethnicity being a characteristic of the individuals who have actually been affected by racism, and make it more about the effects of racism. Kof?
Yeah, no, I couldn't agree more, right? I mean, to the point that was just made, ancestry is so important that the studies primarily focused on an ancestry, right? So that's how we know ancestry is important, because they largely concentrated into a certain ancestry in there. To the point again, I think it's really important that while I'm saying that race is certainly a social construct, I think that it might be useful if we think about the other side of the social construct. And that's why I said those that are likely to have experienced systemic racism, because that is actually what race was created to do, was to induce systemic racism, right? So it sort of articulates that really clear and upfront, and that's what we're observing, and puts that lens on it with the interpretation as well.
And again, just based on some of the basic research done on stress, those things have biological effects that can be transmitted across generations. So it's not something to ignore.
I'll just build on that point. I completely agree with everything that Dr. Gerasa and the other panelists coming up had said. And I would encourage all of us to think about how our interdisciplinary collaborations can really help bridge the knowledge that we need to advance the interconnections between the basic translational and clinical work, enhancing the way that we are measuring environment to tap into these constructs that are shaping disparities.
We've had a number of comments and questions that relate to what are the questions we should be asking. And Raul touched upon that in terms of variables to include in a study. I'm wondering if the way each of you think of it, if you have in your mind a set of default questions that must always be asked unless there is reasonable evidence that they're not important. Do you have a set of default questions that you ask in every study that you do, or perhaps, yeah, if that's not too much, but perhaps think, let us, because you're from 3 different disciplines. So if you could enlighten us on the sort of default questions you tend to ask, that would be great.
Well, I'll tell you that what has generally been done in the very narrow, small studies that I've been a part of focusing on neurocognitive functioning is the typical checklist. We have these tables from NIDA that we got to fill in and report that use kind of the US Census type of classification. So we have to ask our participants those questions. Over the years, in my experience, we've been adding things like level of mother's education, income, in these small studies. With ABCD, we've had the opportunity to expand well beyond that. And that's where we've brought in the measures of discrimination, the geolocation data, all of these other amalgamation of things that we know impact folks in adverse ways and are not distributed equally among folks fitting in various races within the US. So I do not have a standard set of questions. I think that ABCD does have the opportunity, given the breadth of variables that we're currently collecting, to perhaps be a leader in showing how these variables can be much more informative and important than just looking at race.
And if I can add this, I was going to ask you, Raul, directly whether there was something that we are not asking currently from the ABCD study that could enrich it further. Because as we're going into the process, we're learning from it. And we're about to start a similar study, but initiated in infancy. So we want to actually ensure that what has been most valuable from the ABCD can be translated, albeit for the age of these younger kids, so that we take advantage of the enormous opportunity that the ABCD has had for us to understand overall how socioeconomic factors are influencing the differences between ethnic groups. So what are some of the, from your experience having been so intimately involved with it, where do you think we are missing on the ABCD?
Right now, we're in the process within ABCD of a moment of self-reflection and examination. And I think it's wonderful that all of the ABCD leadership has been supporting this. And then also the NIDA team that works with ABCD is absolutely outstanding and committed in this. So we've actually been reviewing every measure in the protocol for racist and sexist language, asking if there are ways that we can do things better, talking about how we're going to be dealing with the dissemination of information, the data use agreement, et cetera. As you know, Dr. Volkow, the ABCD study is ambitious and it is longitudinal, and we got to keep the youth engaged and the parents engaged. And already we're taking so much of their time. And then imagine with COVID, now we have kids all day in school via Zoom. And now, hey, come do our study for another 4 hours on Zoom after you're done. Like, we're starting to look like school for them, which is a bit of a problem. So there's always this tension in ABCD every year as the various work groups reevaluate what measures to include given the developmental stage that the youth are in, about what stays in the protocol, what goes out, what gets expanded on, what gets reduced, because we only have so much time and resources. So the culture and environment work group, which has some of these items, we're limited to about 18 minutes of time with asking questions of the parent and another, about 15 of the youth. So I think resources for the geolocation data and data that can be collected passively in the background can greatly expand the amount of data that we have for the youth without having to burden the families even more.
I want to allow others to jump in. Oh, go ahead, Nora.
No, I was going to say I agree wholeheartedly, and I think that this has become evident in some of the studies that have already been reported on the basis of neighborhood deprivation, for example, and that's a measure that we don't necessarily need to take from the individual himself or herself, but it may be there in that space that we may be able also to expand. So we have measures of school performance, but they have to be normalized with respect to, you cannot translate from one school to the other. So I think that if you, in thinking about it, trying to figure out what would be desirable would be very valuable, because then we can try to tackle what is it that we need to do to make ABCD able to get this information?
I want to allow others to jump in before we leave the conversation, just about default questions or the sort of the considerations that always go into your research that you'd like others to be aware of. Natalie?
Sure. So I would say that, when we're talking about measuring culture or social environment, the opportunities are endless. And I think that it really will depend on the goals of a given study, because as Raul says, there's, resources are constrained. We can only expect so much of our participants. When we can have the opportunity to link information that may exist elsewhere, such as from census or administrative data, that's great because it doesn't present participant burden. But when it comes to questionnaires, there's so many options out there. I actually think ABCD did a very good job being familiar with the protocol. And I think, it's amazing that there will be an infant cohort starting because, in my view, as a child development person, what is missing is, what happened in the earliest phase of life. So I just think it's fantastic that NIDA is planning, a study to start earlier. And I would say that something that, often doesn't get as much attention as may be ideal that some of these future cohorts could be thinking about are the enrichment or those protective factors that, are good for all kids, but particularly may be helpful for those who may be experiencing other types of environmental or social or physical challenges.
Bob, do you have a last word on this?
Yeah, and I certainly work with mice primarily, so my framing is a little bit different. But I will add that I think there's an incredible utility to not only thinking about risk, but resilience as well and protective factors, right? It may be the case that if we start to frame our enterprises around protective factors, we may find things that can be generalizable in a causal way to protect individuals against the broader impacts of things like racism that are generally applied across groups and populations. So yeah, I second the idea of protective factors and resilience.
That also addresses a number of questions we got in the Q&A about how to basically apply what we're talking about to prevention, not just treatment or precision medicine. I'm going to ask Will how I'm doing on time because I'm terrible at that.
We're good. We've got about 5 minutes.
Oh, excellent. Thank you. In terms of getting back to some of the ideas from yesterday about sort of a systems perspective on some of the research we do, I think a lot, for example, one might say that a lot of the research or the perspective coming from, say, basic research we do in non-human animals tends to focus on group differences. Basically, we create groups on the factors that we want to study and or individual differences, basically individual differences in response to the factors that we've imposed on the animals. But that doesn't actually take into account large systems and the role they play in human health. And I'm wondering what your thoughts are on how we can, it's almost a paradigm shift to think about that. And I'm wondering how you think maybe we can do that in the realms of science that don't typically think about social systems.
I really dislike silence, so I'm just gonna jump in. So I think this has to come from the top down and from the bottom up at the same time. I have so many wonderful colleagues with the best intentions in the world. It's just not part of the way we think. And it hasn't been part of the way that I came up thinking. As a clinical neuropsychologist, the focus is on, all right, eliminating variants, don't misclassify people, don't call people impaired. Let's try not to think about race, let's kind of make it through this. There is a paradigm shift happening. And hopefully more and more colleagues will come along and see the importance of this. So having the great role models, having a talk like this put together, seeing Dr. Volkoff's commitment to this, having leaders across our studies commit to making this at least something that folks are aware of and reflect on, seems critical. So this is where some of this training comes in that I think more and more of us are experiencing in our universities, in our different systems, because I think what's missing more than anything is just folks having enough time to educate themselves about things that they typically haven't had to learn about or educate themselves about in the past. So, you know, more knowledge and more learning.
Thank you. Natalie, could you jump in?
Well, this is a hard question for me because I'm an epidemiologist working with population-based human data. And so systems very much are part of the type of research, the questions that I might design and think about. I think that there is a major challenge for, even with human research, we may know theoretically that our participants are existing within these systems, but empirically it can be very difficult to connect everything that we may know are part of the patterns that we may describe with these limited data points that are available to us. So I think that we all have a long way to go, whether we're working with mice or working with human data, to really link together these multiple factors that are driving health and health disparities.
Thanks.
Yeah, and I'll simply say systems are super important, but we aren't always aware of how much complexity we need in the system to solve the problem or the challenge that is in front of us. Certainly, it is indeed the case that mice are not humans. I tried to give my mouse psychotherapy and it didn't work very well. The mouse didn't listen. I was on the metro once and I saw a mouse, running down the side. It looked nothing like my mouse in the lab. It moved much faster and seemed a lot more dangerous. So I can really appreciate the difference, right? A model is a model, right? A paper airplane could be a really great model for lift and drag. It might be a terrible model for jet propulsion systems or fuel consumption, right? So I think we really have to understand what our question is, what we're interested in, and then the best way to use a model to target that. And some things we might, it might not be the right model to answer that question. I think that's something that I've appreciated, that there are a healthy group of basic science researchers who certainly appreciate how to use a model in the right way that will ultimately teach us about what we're seeing in the clinic. And certainly there are things that we can learn about brain cells and the electricity that goes back and forth across their membranes that are absolutely applicable to the work that we see being done in humans as well.
Thanks. I'll wrap up here, and I'll just wrap it up by saying thanks to all the panelists, thanks to the speakers earlier today, and it's been a great discussion. Thank you, and I'll hand it back to Will.
Holly, I will second that. This was a phenomenal session, thought-provoking, and I think it really got to the core of the responsible integration into basic science. I mean, I think it was just absolutely fantastic. So I want to thank all the panelists. I want to thank Holly for that great moderated session. And so we will now break and resume at 3:20.
Okay, so it's now 3:20, at least according to my computer clock. So we're going to get started on the last session. So what we are going to do is we will have the last panel, and then we will have a few final words that Will and we will have just to close the session at the end of the panel. So I want to thank all the panelists for agreeing to participate. This last panel, or this discussion session, is priorities for understanding the biological impacts of racism and how basic science research can advance efforts toward racial equity in health. And so I want to introduce Mehdi Farrukhia, who has very kindly agreed to be moderator. So Mehdi, whenever you're ready, the floor is yours.
Sure. Thank you, Carlos, and good afternoon, everyone. Thanks for sticking around. Welcome to the last panel of this very exciting two-day symposium. As Carlos mentioned, I'll be moderating the session. Just a brief introduction. I'm a staff scientist at the NIH Intramural program in the Translational Addiction Medicine branch. So I come from a little bit different world from most of the previous moderators. I'm in the intramural program doing research at the NIH, and we have 4 panelists. I want to give them as much time as possible, so I'll do a very brief, very brief introduction, just their names and their affiliations, and I'm sure you can read about their work on their websites and bios. So we have Dr. Gene Brody, that you heard from him this morning. He's a professor of child and family development and director of Center for Family Research at the University of Georgia. And then we have Dr. Tali Baram, who is a professor of pediatrics, anatomy, and neurobiology and neurology at the University of California in Irvine. We have Dr. Jean-Luc Cadet from Intramural Research Program. He's a senior investigator at the NIH IRP. And finally, we have Dr. Rohan Palmer, who is assistant professor of psychology at Emory University. And as Carlos mentioned, I think this panel builds really nicely on the previous panel. There will probably be some overlap, but here what we really want to focus on is the priorities for understanding the biological impacts of racism and how basic research can help us advance the efforts toward racial equity. With that, we did have some questions from the panelists, so I'm going to start with the first question, and these are just umbrella questions to brainstorm. And again, I'll try to speak as least as possible and let the panelists talk. So the first question that we had, we brought to the panelists is, how can we more effectively study the biological impacts of racism and whether or how they may play a role in substance use and addiction? And again, this may tie to some of our other questions, but for the first question, I know Dr. Baram had some slides that she wanted to start with. And then
I'll take that, Mehdi. Thank you so very much. I think one can really sort of rephrase the question a little bit is, what is the role of basic science in, particularly as it is defined in terms of the use of perhaps experimental animal models, in really helping us understand the biological impact of racism on the potential, the vulnerability for substance use disorder? Now, this has been an amazing meeting, and we've heard quite a bit through the use of anthropology and economics and other remarkable disciplines about the societal aspects of racism. Perhaps we should bring up evolutionary biology to really introduce the biological aspect that we're dealing with here. So throughout evolution, I think going all the way from tadpoles to humans, when there is adversity, such as that which is generated by racism, be it emotional or biological, there is a response and adaptation. And that response and adaptation is crucial for survival, but also has a price. Perhaps an example would be that, you know, there is the spadefoot toad or whatever that lives in puddles in the desert. And when the puddles begin to dry up, there is a growth acceleration of the tadpoles, and they really mature, and they can survive and overcome and become toads and deal with that stressful adverse situation. But there's a price to pay. They're smaller, they don't reproduce very well, and they die. And that die younger. And that principle, I think, really tells us something about the biology of the consequences of racism. And of course, there is a whole other complexity of where it's coming from, avoiding it, eliminating it, mitigating it. And I think we need to embrace the complexity at the same time that we really look at the biological consequences that we need to deal with and address and study and understand. And that may be the power of basic science. I will now really perhaps share the screen and really aim to discuss a little bit about the kind of questions that we can ask using basic science and the kind of questions that we should not ask. Clearly going into either the human psyche or trying to understand systematic and systemic and societal factors using experimental animals may not be the most appropriate question to ask. But there are questions that we must understand, that we need to understand. And particularly that may pertain to the consequences and the means by which an individual at whatever stage of life perceives the emotional and or biological and physical aspects of feeling unwanted or being physically or emotionally attacked, if you will. So if we want to understand how early life stress or adversity, as defined by our system, impacts the brain, an important time to focus on is early in life, because this is during sensitive periods that were well described for different types of brains and brain circuits and modalities and region. This is a particularly important time. And we need to realize here that there is no dichotomy between biology, if you will, and society, because brain maturation in humans is really impacted by both. Physical and emotional adversity, which is what we're seeing here in this Hispanic kid at the border, is one way in which the brain is being impacted together with the rest of the body. And that may lead to vulnerability to many different disorders, potentially also to substance use. I mean, again, this type of adversity has been linked to diabetes, obesity, what have you. And the question here on the table is whether it affects the vulnerability, the propensity to substance use disorders as well. There is also the fact that the most proximate source of sensory signals to the developing brain comes in from the caretaker, oftentimes the mother. And the adversity and the stress and the other consequences of dealing with socioeconomical difficulties, racism and what have you, really impact the behavior, the overall life of the caretaker, and these may very well get transmitted to the child. And the question is, do they? Is this important? Do we need to study it? One can model it in experimental animals, and again, what I'm showing you here is simulated poverty, when you have a poor resource environment in which you can generate and grow individuals of whatever background you like for genetics, or eliminate that, simply impose that. But then when you look at the behavior of the parent, in this particular case in the rodent, it's the mother, that is imposed upon, that is generated through that, if you will, societal situation or simulated poverty, you see massive effects on the behavior of the dam and the nurturing that the dam will provide to the pups. In this particular case, the behavior is fragmented and unpredictable. And the outcome that one can see if you only have a of that nature, or if you have also the dam, the behavior consequences, you can see that you're going to get a different type of outcome, particularly as it pertains to substance use disorder. So animal and basic science can provide models that can answer some questions. They also allow you to go into the brain and not deal with it as a black box, but really understand how things happen. And of course, complex behaviors such as pleasure and reward and the propensity perhaps for substance use disorder are driven by brain circuits. Sorry. And we can visualize structurally differences in brain circuits that are directly causally related to that particular type of early life adversity that I described to you. And whereas we see here some structural changes that you can see in brain circuits, we can also learn from Dr. Zirasa's work that there are functional differences in electrical circuits within the brain. So this is a question that we can address, namely, what might be the mechanism at molecular, at gene level, or epigenetic level, at cellular level, at network level, that the consequences of racism and other adversities early in life have, and how they get propagated and how they get, I would say, imprinted, or how they generate potential issues at these different levels, including behaviors that may be analogous to those involved in substance use disorders. A second question that I think we really do need to look at, and I was a little surprised and perhaps missed in this tremendous conference, is how do stress and adversity generated by a variety of issues, and racism is prominent among them, impact the brain specifically and differentially in men and women, in male and female, in boys and girls, and perhaps promotes sex-dependent vulnerability to substance use disorder. And this is a critically important question because we know from a variety of epidemiological and other types of studies throughout the world that the massive sex differences among users of different substances, including heroin and including in the current opioid epidemic. And it was mentioned earlier in this meeting that whereas progress is being made in some population, particularly white population, in mitigating the opioid crisis, such progress may not be being made as much in other populations. So how do we get to that? Clearly, we're dealing with societal issues that distinguish men and women, but there's also fundamental differences in the female and the male brain. And indeed, if we plot that, if we model that, and if we look at how early life adversity and what I've shown you influences selectively and specifically and differentially the male and the female brain, we replicate what we see in the epidemiological human studies, and we begin to get a sense into mechanism. So what I'm showing you here, that after the early life adversity in a female brain, although there's not a particularly increased propensity to engage in, or the amount of opioids that can be used, there is a very significant increase in the time to extinction. And when the opportunity arises by cues to engage again, to reinstate the use of opioid drugs, again, there is a marked increase in the interaction of early life adversity and this type of cues, such that exposure to early life adversity increases the probability of reinstatement. All of that is seen in female and not seen in male. Again, suggesting that the experimental approach, the animal models, can replicate or at least create a situation that is similar to the human situation, thus providing massively powerful tools to use modern technology and try to understand that. So these are the kind of questions that we can ask. They're clearly questions that we cannot ask, but we can look at causality. And I think that is something that controlled, rapid experimental models can do, and we can understand the brain mechanism. So the point I'm trying to make is that the complexity of the impact of racism and its consequences on substance use disorder is immense. There is no way or a single way in which we're going to get into it. I think the idea is that we need to do all of them, and we need to choose the questions that are appropriate for each model and absolutely embrace the complexity and move forward from the societal and biological perspectives at the same time, and come up with therapies that the molecular approaches and the circuit approaches in animal models can share with us. So that was what I was hoping to share with the group.
Great, thank you so much. That was a fantastic start, I think, building a really great foundation for our conversation. Does any, I don't have any particular order, any of the other panelists want to jump in, give their opinion, or respond to what Dr. Brown mentioned?
So I want to address what she mentioned. Some of it covered some of the stuff that I wanted to say, but I think the first step towards doing anything is to recognize that racism does have an impact. I mean, we have to stop obfuscating about the fact that racism is a real issue, and that just to bring back to what we're talking about, in that ancestry, African ancestry does matter in the United States. There's no avoiding that fact. So once we come to that point, we can then start asking questions like the one Dr. Brown brought in her slide presentation about how does racism impact biological markers? And I was very enthusiastic about Dr. Brody's talk earlier, because that also covers some of the issues that I have in my little brief, my brief comments here. So since racism is a stress disorder, we can then focus on neurohormonal effects that are being seen in stress-induced disorders, okay? So stress causes physical and psychological disease, including substance use disorders. So that those are known facts. So there's no reason to believe that racism is not going to be doing the same thing. So I've recently coined this racism-induced stress disorders to cover all of the different psychological and medical issues that we can see, including shortened lifespan. One of the previous presenters talked about lifespan in Baltimore City, where I'm located. And you can go from zip code to zip code, and based on who lives there, you can predict that there's going to be, most of the people are going to die on 60. And if you go to the next zip code, the people are living till they're 80, 90. So racism does have an impact. I think when we want to study racism impact on substance abuse, we should include scales that have been validated in studies of racism, like the racial microaggression scales. So those are very important, and I was hoping that a ABCD study would have included some of those. I don't know if they do or not. And I think those type of studies, including scale of microaggression, would help to identify specific changes that racist insult can cause in giving individual. The other thing I should like to mention is that there are differences between acute stress and chronic stress. And I think it's important that we look at those issues when we look at, okay, an acute setting, what does that do to biological markers, and the chronic stress of living in certain area with food deprivation, what does that do to the biological markers that we're thinking of?
Dr. Cadet, if I may just say, first of all, thank you very much for bringing up those issues. I think the complexity, even if we use the word stress as a perhaps mediator for the biological impact of racism, is amazing. Because one thing that is very clear, particularly if you look at specific phases of life and you add sex and you add ancestry, as you said, is that there is no simple thing such as neuroendocrine or a very simple hormonal effect. It looks like as the brain keeps on moving and developing in different trajectories and at different velocities, different ages will add particular processes, different circuits will develop at a different time frame. So even if we consider that as stress sort of as a overarching board, which it may very well be, it employs many, many neurotransmitters and neuromodulators and working on specific circuits and at different time frames and interacting with, again, genes and ancestry, as we heard from Kath. So the complexity is rather large. And I think you're absolutely right also that we need to look at the duration, and not just duration per se, but what the timing of the duration is. So I think your point is well taken, and these are many of the different questions that should be addressed to really understand the impact in humans and in biological models that can then allow you to look at mechanism of the consequences of racism on the brain. So thank you for enriching that.
I'd like to add a cautionary note. In some of our work, we identify young people who were growing up in very challenging circumstances and who nevertheless are resilient, what we call resilient. So they're doing well in school, they're not using drugs, they're very planful, they show lots of future orientation. And as we follow them, they look resilient by all of our conventional markers. But when we start looking at biomarkers underneath, it looks like they're becoming ill. They have high levels of circulating cytokines. They have high levels of epinephrine, norepinephrine. They are more likely to develop type 2 diabetes. And other people in other labs around the nation have found the same thing. So we're replicating this effect. So in essence, what we're finding is a group of people a group of children, and we follow them till they're young adults who look great. We might want our own children to look that way, but because they've had to work so hard and persist and not take their foot off the gas pedal, biologically, they're not looking well. And so we thought, maybe this was a specific effect from 2 of our samples. And subsequent to our work, now in a national sample and 4 other samples from around the country are finding similar things. So my cautionary note is you can get to a health inequality through a different route than simply exposure to social determinants. Now, these people have a single-minded desire to succeed. And because of that, they don't use drugs. But we don't know as they get older and they've had to grapple with racial discrimination on their way to success. They've had to grapple with alienation on their route to success. We don't know how they're going to fare later on and whether or not, despite their lack of drug use, later on they may start to self-medicate. We don't know, but we do know they have lots of cardiometabolic risk factors that they've developed over 10 or 15 years.
Yeah.
Sorry, Juan. Go ahead, Juan.
Yeah, I was just, this is a great point, Jane. And I think one of the things we need to recognize is that, there are multiple factors that are associated with different levels of racism. And the effects that we're trying to get at for the behaviors could affect substance use initiation and development in different ways, to the extent that it might not necessarily be the presentation of substance use, as you point out, Gene, but other forms of psychopathology, perhaps internalizing, which dimensions or versus externalizing that could manifest in young kids. And it could be also that how a person responds to the stressor that is racism could differ depending upon the level of how racism presents to them in their society, because we have different forms of racism. We've got institutionalized racism, personally mediated racism, right? So it can affect how the kind of assumptions about our abilities and whether or not we might adopt those assumptions as an individual. And once that begins to happen, we move on to acceptance. And then we might end up portraying those kinds of behaviors, and we think it's okay to act this way because it's expected of us. And so I think it's important for us to assess, racism, its impact at all these different levels for the individual, for the society, the social cultural environment, and to look at how it could be impacting the progression to initiation and substance use, but also the comorbid behaviors that we know sometimes, predate the onset of substance use.
I'd like to really echo what Rowan said and maybe add another factor, and that is something that we oftentimes don't do in animal studies. And it was really nice to see the cavity that Nettie's. There is, in addition to ancestry and group type differences, there are, of course, individual differences in both the genome and, as Holly Moore brought up, in the epigenome. So that I think how a given individual will respond to the same clear objective racism or not racism related stress, because I think it will happen in both scenarios, is dependent to a large degree on his or her genes. So I think that is something that we need to consider. Again, is there perhaps a genetic commonality to the group of youngsters, Gene, that you found are doing really well but pay the price? And the other aspect that I was hoping to show when we had the caretaker as the buffer to the developing brain is although we think that someone is exposed to the same degree of insult, racism or other, there may not necessarily be, because there is a buffer of aspects of society that protects them. And that again is difficult to model, but feasible. So I think the complexity is really a word that I think we need to turn into almost a mantra in terms of realizing that there are many, many different factors, and we should take them one at a time and then aim to integrate them and take them back to the human study and the clinical studies that are being done. But we have to figure them out, and we have to figure out the mechanism, because there may very well be other different molecular targets and different potential measures of intervention and prevention to address them.
Well, I think I'm glad that Dr. Woody brought up the issue of resilience, because I guess a previous talker talked about that also. In a paper that I wrote a few years ago, I stressed the impact of resilience, because what happens often, I'm been guilty of this myself, is to pathologize Black lives instead of thinking in terms of other factors in that environment can serve as protective factors. So we talk about that. So when we're doing biological studies, and I think when we start seeing changes that are very similar between groups that are resilient and not, we need to start thinking about, okay, what are other markers that can be used to detect resilience and try to connect that to the clinical manifestation that we're seeing. The other thing I think I should bring up is that under this certain condition, and I think Warren brought that up also, is the fact that we can have other psychiatric manifestation of racist insults. It's not just drug use, but depression. There was a nice paper published, I guess last year, showing how successful Black men specifically suffered from major affective disorders who've really been exposed to repeated insult, not acute insult, but repeated racist insult over time. So when you're looking at those population genes, is it possible to actually dissect out substance use from other psychiatric disorders, and use the cytokine markers, which are very important, I think, to maybe they're protected against substance use, but they're not protected against other psychiatric diseases.
Yeah, these are all great points. Taking the discussion to kind of, I think, the next level, especially being the last session, is just wanted to get some thoughts on how really research on biological impacts of racism can advance efforts toward racial equity in health. I think this is a very important topic. So like, let's say we do answer all these questions that will never happen, but then how this knowledge on biological impact of racism can really help us reach or move toward racial equity in general.
I think there's two issues here, and it might be helpful to separate them. One is the issue of NIDA, namely the notion of substance use disorder. Is there a role for racism in substance use disorder? If so, what is it? What is societal, biological? How do they interact? And if we understand it, can we prevent it? The other issue is racial equity and equality and lack of discrimination in the workforce, in the scientific community, and in our view of the world in general, in our systems, in our organization, in this country. And these are two very different ones, two very different questions. I am struggling with trying to understand how basic mechanism of the impact of racism on the brain can take us from the biology to the organizational and systematic way that we need to eliminate racism with. I think there may be a little bit of a disconnect. There are things that basic science can do and things that we should learn from and say, yes, there is a very clear biological effect, a very significant and severe impact on brain development, on the propensity for substance use disorder, and therefore we need to do something to prevent it. But these are separate questions. Nora, I'd love to hear from you about that.
Yeah, no, I've thought about it a lot because I think that basic science provides us a very powerful tool. And I've been obsessed about it with all of the data that's coming up with the ABCD. And to me, we have an answer that is very powerful. And so as you look at the ABCD data, you come to realize that many of the differences in ethnic groups are basically driven by low income, neighborhood deprivation. And then as you look through it, and the work, the elegant work that Jean Brody has been doing, including interventions that prevent, actually by doing a very specific targeted education to parents on how to support their children to actually engage them better in school activities, and how prevention programs that allow those children of underrepresented groups to have high level education so that they, when they grow up, they actually have higher income that protects them, not just themselves for addiction, but it also protects their children. So to me, what I'm looking at in terms of the ABC data is documenting how these adverse social environments is negatively impacting the brain of children and their performance. And thus intervention, why don't we provide a high level, high quality education and the support to do that to children that are coming from these underrepresented groups? I think that this is a targeted intervention that research on basic neuroimaging is speaking loudly to. So that's why immediately I saw that and said, yes, this is a way that we can have a targeted intervention.
But I mean, even that answer, Noah, is not really focusing on the biological markers that we're seeing. So I had a similar answer to that question. The answer is intervention at the level of school. Those kids need to be provided with better food, better school, better education system, and there needs to be anti-racist work done together with their teachers, because the vast majority of those schools are going to school where they have white teachers who might be punishing them much more severely than their white counterparts. The literature is very clear about that. So those kind of intervention, I think, are the ones that NIDA needs to support in order to decrease the prevalence of substance use in that population that we're talking about. But again, I think it's very important that we take into consideration resilience of other kids and not pathologize so much the ancestral background of those children.
Yeah, and I agree, and they are not exclusionary, Jean-Luc. And the other issue to me is that when we start to look at biological differences between individuals, we recognize them as very important. And I think that the example of Kafui that he gave in terms of how between strains of mice, you see differences on how the blood-brain barrier is functioning, allows entry of one drug or the other. So there's tremendous diversity that we need to address in order to personalize interventions. And when we're speaking of African Americans, as everybody knows, the diversity is enormous. So it's just, if we want to understand biology to optimize outcomes for everyone, so I may have higher risk for breast cancer because of this or that gene. It actually has an element that is in some ways distinct to a certain extent of the racism that we observe. The racism is impacting because you're putting it as an enormous stressor that no matter what your biological backgrounds are, it's putting you at a disadvantage. So I'd like to think about interventions where us as a society can do something that changes things. I mean, to me, that's urgent. And based on the data and the work that Jean Brody and others have been doing on what prevention interventions work on their underrepresented groups, I think the issue is how do we implement them and how do we overcome the resistance to implement it and how do we overcome the unwillingness to put resources. So that's where that's why I jump into it, but completely agree, Jean-Luc, that there are other aspects that, and what is the biology of resilience is extraordinarily interesting and important. So I'm not disagreeing, I just think that they are not exclusionary.
And Dr. Brody or Palmer, did you want to comment on this topic?
So, you know, I have a foot in two worlds. One world is I look at etiology and study things like how stressful events like racism impact inflammatory responses and change neural structures. On the other hand, I'm also very interested in translating protective factors that deter drug use and promote competence and development. And so in my world, what I find in etiology and the protective factors in that world, which keep stress, I'll say, from getting under the skin and into the brain, we try to translate into new prevention programming that will improve our existing programming. And so there's this reciprocal influence process. But right now we have prevention programs that are 14 and 10 hours total, some of which are being disseminated in communities around the country and reaching Black families.
Yeah, Dr. Palmer, did you have any comment on that, or should we move to the next topic?
No, I was just sitting here. I love what Jean just said. I mean, I don't particularly work in that space, but we've been in my lab. My students and I have been working to come up with ways that we can empower young people across colleges and universities who we know are at risk, because it's a period for high use of initiation and experimentation. And we think about how do we empower those young people? And then I think at the same time about how do we do that for communities that are even more disadvantaged. And I think it's sort of the same thing here. It's, you know, as Jean said, I think about, and Nora saying, as we identify folks that need, how do we educate them to be interested in this kind of work and empower them to get involved, spread the same knowledge back in their communities. I think there certainly is a place here for community-based participatory research at a level where we can bring these young people so we can shift the culture, get a movement started, because I think we sort of need that grassroots component to this as well.
I'd like to bring up one other issue, and that is we're talking about prevention and the idea of racism, as you said, getting under the skin and into the brain, particularly early in life, that promotes substance use disorders. But I think there's also a strong need for basic science and intervention to look at what the effect of actually using drug is on the brain, on circuitry, and getting into the vicious cycle that we all know and that Nora's written about so well. And I mean, there's clearly genetic differences, there's clearly societal differences, and there's clearly a combination of both in terms of addressing and mitigating drug use in people who already are, helping someone quit, helping someone not get back on drugs. And I don't know that there's much known, in terms of the genetic variables and other variables that determine what the effect of drugs on the brain is at the circuit level, at the molecular and cellular level, and particularly how we translate that into intervention. And I guess that would be an important study area to continue to study. It is being studied, but we do not have the answers.
Yeah, I think that's a good segue to basically one of the next questions that we had. That what do you all think should be the priority of NIDA in terms of basic research science? So again, the topic is mainly looking at basic research, and what do you think basically NIDA should prioritize to address racial inequalities, again, in terms of basic research?
I think I could take that one, but this is very individual bias. So there's evidence, as Dr. Baum mentioned before, that stress causes epigenetic changes, whether it's acute or chronic changes, chronic stress. So I think NIDA should spend a lot of money, maybe more money, on the issue of what does racist induced stress cause to the epigenetic markers in the blood, in other cell systems, and how do those correlate to manifestation of substance use disorders. I mean, I should say that the use of drugs themselves is associated with epigenetic markers changes. So we would need to dissociate the consequences of racism from the consequences of drug use itself. I think that where really collecting brain tissue from large population of African American come in. I mean, this is worked really well. I'm a neurologist and a psychiatrist. Those approaches have worked really well for neurological disorders, multiple sclerosis, Parkinson's disease, Alzheimer's disease. Having a large collection of brains have helped to decipher mechanisms in terms of the basic associated with some brain pathologies. So we could do the same thing in terms of if we know that racism can cause structural changes in humans, okay, it should be possible to follow these humans after and collect their brain after death and correlate clinical manifestations with epigenetic changes, gene expression, protein expression in various brain regions. I mean, Dr. Desara showed that picture of what's being done with the brain at the Lieber Institute in Baltimore. Those are, I think, are the kind of studies that NIDA should support. Thank you.
I think it's a very difficult question. Clearly, I think in the 2 days of this conference, a large number of issues were brought up, and many are really worthy of study. In terms of basic science, again, I completely agree with Jean-Louis that prevention would be an excellent place to start. And therefore, I think the idea of what does the both direct quote unquote stress of due to the developing brain, and what is the mechanism through which environment influences the brain, but then realize the complexity of time of development, which circuits are involved, what behaviors are tested, and really try to get at biological, not just markers, but perhaps at actually biological mechanism. Is there a change in opioid receptors? Is there a change in stress related neurotransmitters? Is there a change in dopaminergic system? And what circuit, at what pathway, at what projection? And really look at the 21st century, 2021, 2030 approaches that will tell us what really happens to specific portions of the brain, to specific circuits by that type of quote unquote emotional as well as physical stressors, and what are the changes that we can then weaponize, that we can then find actionable, that we can target with novel or existing medications to try to intervene. The other aspect that, again, I think should be looked at is really trying to understand, and again, both in human studies, which is being done, but also in experimental models, the intrinsic differences and the plastic differences that are influenced by environment of brain of a female and a male, and really try to understand if we can design approaches that are particularly suitable to women or girls and to men. In that respect, Jean, are you finding very significant sex differences in your work?
You know, we have not, well, let me take that back. Generally, we don't. We found one, I think, very interesting one. We followed up some of our young people who participated in a prevention program at age 11. And at age 25, 14 years later, the prevention program, and we actually hypothesized this, so I yield to you on this. We hypothesized that over time, males would increase their drug use and women would cope more by eating than did men. And the prevention program at age 11 deterred increases in drug use among men and emotional eating among women. So it was efficacious, but on two different behaviors. And I'll just leave it there.
Well, as I've we don't have time to see the same dichotomy in terms of craving of palatable food versus drug use, it exists in animal models as well. So perhaps rather than going through a 14-hour massive intervention, if you can find the magic bullet that will prevent that, that would be a very reasonable endeavor to indulge in for NIDA.
Yeah, for sure. One of the things that has become more clear for me is that, to coin a phrase, it takes a village to do this work right. So I have wonderful, wonderful colleagues, some of whom are neuroscientists, some of whom are immunologists and have great laboratories. I have some colleagues who are psychiatric geneticists, some who are health psychologists, and it takes all of these points of view together to get at different levels of analysis from which you can start to paint a picture that really does inform prevention.
I had a question as we were discussing all of these issues, because there's something that I have certainly in my brain. It's been extraordinarily sensitive in our field in substance use, where we show one of the most devastating components of racism, and that is incarceration of African Americans. And this is so very closely linked and tied with drugs. And so as I was thinking about the notion on how do we use basic science and knowledge to actually advance and overcome adversity for minorities, I mean, one of the ways that we can have the highest impact is to actually bring forward data and knowledge that can help inform policy. And in this respect, for example, investigating how adverse, horrifically adverse, it is to isolate someone through prison or jail in general, but more if this is a teenager or a young person, jeopardizing their outcomes into the future, and understanding, actually one could even do this, in animal models, how disruptive this is as a practice. So I think that that's another aspect that we cannot ignore when we're discussing issues of racial differences as it relates to outcomes on drug taking, but also if you're taking drugs, you all end up in jail or prison, and from there on, your trajectories change forever. So I think this is another component that we in the field need to very actively, proactively address.
Yeah, Dr. Palmer, did you have any comments on priorities?
Yeah, kind of talked to some of the people in my group, and we came up with some ideas. And one of these was to, as Nora was suggesting, careful monitoring of data and substance use morbidity and mortality in these disproportionately afforded groups that are at risk. And I mentioned earlier just sort of this idea of more emphasis on community-based partnership type approaches, so that we can assess the needs in these types of communities and think about developing educational resources that Jean and others are already testing, and applying them to develop policies and infrastructure that we can use to combat systemic forms of racism and repression that we know create these health outcomes in these groups. Another idea was to support research related to implicit bias training, right? And so, to the extent that if we can look at implicit bias in the treatment and maintenance of substance use disorders, and how implicit bias training can actually impact clinicians and their effectiveness, right? So this idea of, if we can teach them about the history of abuse within the healthcare system, that has actually led to patients' mistrust or distrust of the healthcare system. Would that kind of training actually have an impact and lead to improvement in these disparities? And the third was this idea of using both qualitative and quantitative methods to monitor the endemics of racial bias. So this idea of, can we integrate assessment of racial bias in our core data systems? I'm not sure how popular this might be, but can we call upon OMB, for example, to require that every agency collect racial data? And as a consequence of that, help to create new data systems that can monitor the impact of racial bias. I think one of the things for me as a statistical geneticist is about being careful about how measures of ancestry might get used in genomic studies to infer risk. And I think it's very important that if one wants to do that, that it's done in the context of the impact of racism on the individual as well as how a person might self-identify. And we can't begin to ask those kinds of questions or begin to even interpret things like genetic scores and so on and so forth without a good data. There's lots of data.
Great, yeah, thanks. Really, really great points. So we have almost 7 or 8 minutes left. I just wanted to open the floor, give each panelist 1 or 2 minutes if they wanted to mention any point that they didn't get the chance, or any other thing. This is the last session, so reflection on other sessions that we had, or the questions that we have, or any other topic you wanted to bring up.
I'd like to bring up the issue of transdisciplinary. So again, everything I know about drug addiction is from a collaboration with Steve Muller and other people who are really experts in the field. And we all bring different expertise and background to the pot. I think, Jean, you just mentioned that it takes a village. And I think one thing that I would recommend that NIDA embraces, again, perhaps like the Conti Center in NIMH, is the ability to create in the same place multidisciplinary teams that go and not necessarily dichotomize into animal research and human research, but as we do in the Conti Center, we actually do both. We can take a question across species. We can take a question across disciplines. We can look at the consequences, for example, as you said, John Lude, of early life adversity on the single cell, on the epigenome, on the single cell transcriptome. We then go and look at the interaction between cells. We then go map circuits. We then look at behavior. We then look at a variety of different behaviors, both cognitive, emotional, and those related and non-related to substance use, to really get a phenotype, whether it's an animal or a human. We can then carry the results of the question that arose in the human back into the human and look at intervention. Just to give a very simple example, I mean, we all had now witnessing what COVID-19 is doing to everybody. In our case, it was preschoolers. We tested the hypothesis that really preservation of family support might be a potential intervention. And indeed, in a study that just came out, it appears to be the case that preservation of family routines was the most potent discriminant between a variety of psychological measures in preschoolers exposed to the stress of COVID and those who did not. Again, bringing together psychology, animal studies, because we predicted it from the animal model, epidemiology, and an opportunity arose by a terrible pandemic. So to go back from this example to the overall story, I think that we are tackling a very complex entity. And I think we clearly need to move forward with intervention, but also need to really understand the impact on the brain in substance use disorders and others. So bringing together teams that have many different disciplines that can look at things and from the computational approach, from the epidemiological approach, and from the basic science approach, and suggesting that that be done across species may be an important way to go.
So I would like to continue that thought to include the fact that by doing things in a multidisciplinary way, we can actually train the next scientists especially by increasing the number of African-American young scientists who come into the field, because those investigators will have more easier time interacting with communities such like one in Baltimore, because they'll, just like this, talk about the fact that there are people within his family who are suffering from psychiatric disorder that prompted him to follow MD, PhD and go into psychiatry to study this thing. So by not only doing research related to racism and epigenetic changes, but we can train people who want to do that their business going forward.
Dr. Brody, any other last word, any other thoughts?
Yes, I want to thank NIDA for putting all this together. I think you were able to convene people who otherwise wouldn't talk to one another because they're either too busy or it wasn't disciplinary enough. And I think this was very important for the field, so I want to thank you.
I'll echo that very much so.
Same, thank you.
Oh, the thank you goes to you all. Dr. Palmer, any other last word, or just giving you?
No, just very grateful to be here. And
Great. Well, thank you all so much. With that, I know we are on time, so I'll turn it over to Will and Carlos.
Great. Thank you, Mehdi. Thank you, panel. That was a fantastic discussion. A lot to learn from that. I think there was a lot of recommendations, clear recommendations on points to consider as we move the research agenda forward. I think there are many aspects of the discussion over the last 2 days that we need to really sit with, think about how best to integrate these recommendations and moving it forward in a significant way. The recommendations, I would say, that took place over the last 2 days were not incremental in any way, but these were very transformative, bold, thought-provoking ways of moving things forward from a prevention standpoint all the way through treatment. And that is the types of discussion that we hope would come from this meeting. And I think in every sense of the way has been very successful in that regard. So I tend to look at this as a marathon. This is the first leg of the race in terms of outlining what some of those key priorities are and where do we go from here. I think we have some clear directions on how to move this forward in that way. So I want to thank each and every one of you as presenters, as discussants, and the moderators as well, and my co-chair, obviously, for this meeting. This has been a very important meeting. Carlos, did you want to say a few words before we close the meeting today?
Yeah, no, I'm also very, very grateful to all of you. I mean, it's been really exceptional 2 days with so many years. I mean, I maybe because I'm less athletic than Will, rather than a marathon, I feel that this has been like going to a restaurant and getting the tasting menu, no? We have sort of sampled a few of the things that are in the menu, but I hope that we can come back and have a, maybe a fuller meal or several meals, and we can advance the science. I mean, there are so many topics that have been discussed today, from the fallacy of the biological fallacy of race to really the biological effects of racism, ideas for interventions, ideas on how to study the consequences of racism. So, I mean, there's like, we could stay here and sort of spend the whole day to continue to discuss what we have done. But I think we have opened the door for many discussions. I am again very grateful for all of you for being engaged. I have been monitoring a little bit also the Q&A chat, and a lot of contributions both yesterday and today. So the richness is amazing. Before we close, I see that Nora is still connected. So I wanted Nora to see if you wanted to say any words to officially close the meeting.
Well, first of all, thank to everyone. And I think that the presentations are the words that we should actually keep on resonating in our brains. I mean, obviously, there are a lot of unanswered questions, and there's an enormous amount of opportunities and challenges And at the same time, to me, is as we come along with what should be the priorities and how to implement them and how can NIDA help, the next component, I mean, how can we apply this knowledge to address the crisis of disparities that we lived and we have lived for, I don't know how long, but that the COVID pandemic is making so obvious, and that it has generated a momentum that we don't want to miss the opportunity, because I think that now if we move strategically, there could be an enormous advances, and you already see it in the excitement and commitment of people about the importance of this topic and ways to change it. So I thank you all for your commitment to the idea of racial justice and equality for all, and death to discrimination. Of course, this sounds so nice that it's actually say, but it is. I mean, we are driven by a mission, and to have a mission that is so basic to the ideal of how we should be, I think should give us the strength and the energy and the commitment and the resilience to continue doing it despite all of the challenges and complexities that are thrown at us. So I want to thank everyone for this. Thanks a lot. And thanks, Carlos and Will. This was really terrific.
Thank you, Nora. And I want to go on record for thanking you for your support and leading this effort and your vision. People look to NIDA for guidance and what is NIDA doing in this, and I want to thank you for that. I will also be remiss if I didn't thank the Research Gaps and Opportunity Committee, as well as the contractors as well. So thank you, everyone. This was an important meeting, and thank you all.
Okay, I think we have to say goodbye.
Goodbye.
Bye-bye.
Bye.
Bye, everyone.